Hello, everyone. Welcome back to Undoctrinate Yourself. Today, I'm sitting down with the incredible Kevin McKernan. If you don't know his work, you need to follow him on X. And of course, in the show notes, I'll give you all the information, places you can find him, things he's working on. But Kevin is a molecular biologist by training, has a very interesting background, and actually ended up defecting from academia, which was a sentiment that I probably kind of stuffed down multiple times throughout my PhD. And he has a great success story. So I'm really excited to talk about his training and the opportunities that arose. And with the recent issues with the Department of Education and the bills being passed there, I think it's an important conversation to have. We were chatting about that before we came on, because there's this idea that, people are outraged at, this department getting cut. Meanwhile, we were talking like the market isn't driving PhDs. And so what you end up having is a bunch of PhDs that can't find jobs essentially. And the degree isn't really worth that much. But anyway, we'll talk about that in a little bit. First of all, just welcome onto the show. I'm so happy to have you.
Thank you for having me, Alexis. I've enjoyed your work as well. I've been following your stuff with Jack Cruz and a few others. So I usually go on podcasts where I can learn something. So I'm high expectations here.
Great. Yeah. And I definitely want to make sure we talk about the blockchain stuff as well. And I have my Bitcoin sign behind me. So we'll definitely touch on that at some point along the line. But let's talk about your training a bit and how you got interested in genetics, molecular biology, what your kind of winding path was to where you are now, and then we'll get into some of the recent developments.
Yeah, so I had an early start. I got really lucky. I got a job on the Human Genome Project back in when was that, 96. And it was one of those scenarios where it was at MIT, and so a lot of people there that were running it were from MIT, but a few of them were running the research and development program there. I think they felt like they had to leave MIT. It was one of those things like they graduated and they were still at MIT, and so they took off and handed me the reins, completely unqualified, and I had to kind of scramble to figure out, okay, how do I fit in here? I'm not a PhD in molecular biology, let alone am I qualified to be running an R&D program on the Human Genome Project. But thankfully, there were a lot of people there that just put faith in the fact that I was a hard worker and eager to learn, and I filled a void. And then over time, that program grew tremendously. A lot of funding from Francis Collins, ironically, and I ended up learning on the job. And throughout that program, I kept getting tempted to go into a PhD program. Everyone kept telling me, "Well, you're doing all this exciting work. You should go get a PhD in this exciting work." And I started down that road. Applied to a program out at Seattle from Lee Hood. If you don't know Lee Hood, he's one of these like Elon Musk like characters in the biotech space, the genomic space. He invented some of the first DNA sequencers and synthesizers, and just he's got a great repertoire of bringing out really incredible technology into the field. I then deferred from that program when there was a family health issue here, thinking if I moved across country from Boston to Seattle during a PhD, I may not get back for 6 years, and that was a long time. And so I tried to get Eric Lander helped me get a position at MIT as a special student where I could start taking classes toward a PhD program. And as I started taking those classes, I realized they were like years behind what we were actually doing on the Human Genome Project. So I'm like, my day job's paying me. I've got to leave my day job to start paying them to get maybe a stipend where I can live as a student. Eventually, I ended up finding an opportunity to start a company and just said, you know what? To hell with a PhD. I'm going to go and start a company, and I'll be a really insecure 23-year-old CSO. What was that then? I don't know. It was 23, but it was 19. It was 2000, so I was probably 27 at the time. And once that company got going, it quickly became profitable. We started selling off portions of it to different larger companies, and we started building some DNA sequencers. So that's kind of the compressed version of my history there. Where I toyed with doing the PhD, but it was one of those scenarios which I think you'll often find if you're in a cutting edge field. The PhD programs, I would imagine, like maybe the PhD programs in AI today are probably or blockchain are probably years behind what you could learn if you're just working in the actual industry itself.
Totally.
Uh,
Yeah. I feel like
yeah.
academia tends to be on the lagging end of things. And I mean, I can just speak personally as, I finished my PhD at Princeton and I saw a lot of my colleagues and peers, vying for postdoc positions. And then from the postdoc, you're like, basically it's like a cutthroat situation to try to get a faculty position. Or if you can't do that, then you go into pharma or biotech or something like that. And there's a very small amount of faculty positions relative to the amount of students that actually want to become faculty. Like there's a ton of people who like aspire to, you know, have their own lab and be a PI, and there's just not enough positions for them. And that kind of ties into the conversation we had briefly before we started recording, that the 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 PhD programs are not actually market driven. Do you want to talk about that a little bit? I think it's relevant given the whole DOE situation.
Yeah, so it's a pricing signal thing, right? So how do the universities know what PhDs they're producing to satisfy what the market's building? And that always is a bit of a chicken and egg issue. So they can overproduce the wrong types of specialties. If they happen to be academically in vogue, we might have an excess of gender study PhDs right now, which have no real market position. Maybe they did a year or two ago when they wanted to have a lot of this DEI positions in schools, but those are getting quickly eliminated because it was more of a zeitgeist than a market need. And so that can happen in science too, where we can funnel people into Alzheimer's research, all thinking about tau protein in ApoE, and we find out 20 years later that that was maybe a symptom, not an actual cause of the disease. I remember working at Emory. When I first did DNA sequencing was actually at Emory University, radioactively, and we were sequencing serotonin receptors because they were trying to fit serotonin selective reuptake inhibitors. Now, the whole SSRI craze was going on from in 91 to 95. And now we're looking back at that, realizing that's probably an antiquated model of depression, and maybe those drugs are have suicidal ideation, and there's other things you could take from mushrooms that would probably do a better job. So sometimes that type of fiat funding makes us go down rabbit holes that don't have a price signal on them, and I think this derives from printing money. And when we print money and we hand it out to universities through a grant system, Anthony Fauci can decide that HIV is a significant threat to the world, and we have to pile all this money into AZT, right? When AZT is in fact, a toxic cancer drug, and that wasn't really the right thing. It was kind of declared from on high that he is the science, and that's what's going to get funded. And so huge rabbit holes get drilled by academic research that are dictated by $42 billion coming from on top. And that's not necessarily the market deciding that that's a right financial investment. That's a few people at NIH that have a disproportionate amount of control over the expansion of the money supply. So I think it's a good thing what they're doing is they're trying to tear some of that down, and everyone's screaming because they think it's going to be the end of the world. The Department of Education, if you get rid of that, we're going to go back to being cave people. But reality is when I speak to teachers, they're like, the Department of Education does nothing for them. It just makes more administrative cost. And then the admin budget is so large over what the teachers get that they find themselves doing all these administrative assessments and behavioral plans and what have you that don't benefit the students. If you look at the administrative bloat on universities in the last like 2 decades, you'll see why college has gotten so damn expensive. That means if you're going to go into a PhD program, or to med school, you're going to be building up almost $500,000 worth of debt that you've got to figure out how to pay off. And the price of the price of your salaries coming out of those programs isn't necessarily escalating with the price of the cost of the education. So there's a lot. A lot of it comes back to monetary policy, and it's hard for people to see that. But when they're flooding money into systems like this, it distorts everything. Everyone feels like they have to go get a college degree now, otherwise they can't get a job when they get out. But I don't think that's going to be true in 5 years. In 5 years, I think a lot of these intellectual jobs, pushing spreadsheets and doing market reports, are all going to be done by AI, and the only jobs that might be available are people who actually use their hands. That are involved in maybe electricians and plumbers and actual trade school types of professions, which are arguably cheaper educations to actually acquire today. So it's a complicated thing to unwind if you don't know about the money system.
Yeah,
I
the
mean,
blow is a really big issue, and I mean, as like a personal anecdote, even if you get paid a stipend from a university, so like I was making $3,000 a month on my PhD, to live around Princeton for that rate is not really doable without going into some form of debt, and then you're there for 5.5, 6 years or whatever. You're accumulating debt over that time, and then, you have to go get a job, and maybe, if you're getting a postdoc, it's like not much better. You maybe get $4,000 a month. So then the debt is still building, and then, oh, you get a wonderful faculty position, and you start at like $120,000, which as an undergrad sounds like a whole lot of money, and then by the time you have all
they
this debt,
starve
it's like,
you.
this is nothing.
Yeah, yeah, right. Yeah, yeah.
Yeah.
So I mean, I don't know that it'll be interesting to see when they do when they peel some of this stuff back. Then the universities have to decide, okay, how much of our endowment are we like? How much do we really love teaching students? Because right now they're doing it because they can fund them off of some grants. They subsidize some of this type of activity through grant work, and when that grant work dries up, they're going to have to ask, do we use our endowment for this? Do we, or do we only focus on building PhDs we know we can place in the marketplace and can hopefully contribute to the school through donations when they're successful, right? I mean that should be the model, is that you build your endowments with successful alumni who contribute back to the school, thinking it was you want to pass it on to the next generation. But I suspect they're going to have to slim down the number of PhD students if the grant system needs to get overhauled. I mean, the most recent overhaul I think they were screaming about was that the overhead in the grants they want to put back to 15%. All right, that's not unreasonable. We've been involved in some of those grants, and yes, some of the schools like Harvard and MIT have really high overheads. They rake probably 60 to 65% of the money off the grant and put it into their institution for supporting what they call, keeping the lights on.
Yeah.
Um, but they've now gone in and said, no, we're only going to put forward like 15% for overhead. The rest you guys have to figure out. And now that's making them cut the PhDs. But what they really should be looking at is, why is our overhead so high?
Yeah.
Like, the PhDs might actually make inventions, and maybe you'll get patent royalties, or maybe they'll start companies. But the reality is the universities just have a really massive overhead they don't need, and their first reaction is to cut the PhDs, but they should probably be cutting their administrative staff that has bloated over the last 20 years, and see if they can get by with a more streamlined operation.
Yeah, I mean, it seems like most of the bloat is really comes back to middlemen of some sort that are like kind of superfluous, but they're kind of these fake jobs that are just adding to this overhead, like you're saying, but they're not really serving a real meaningful function. And I mean, that's like all insurance companies are basically like this. A lot of the administrative people, and it's just like it's the illusion of a job, but you're not actually an essential worker in any meaningful way.
Yeah.
Yeah.
It'll be interesting to see what happens. I don't think it's going to be doom and gloom like everyone's predicting. It's the end of education. And so I think having them put some pressure on there is going to be, we'll have some interesting outcomes. I think that the very resourceful universities will figure this out and be like, okay, we have got to trim our overhead if we really want the rewards of research, which they claim it creates jobs, it creates companies, it creates IP. Then they're not going to get that out of the people pushing paper around, making sure that they all have diversity, equity, and inclusion in every step of the corridor. That stuff's going to have to go away in order for them to get back to the basics. But um, there will be no shortage of screaming on the way there, I think.
Yeah, I think it's mostly because we have this warped sense of reality that's been going on for the past few decades where it's like we think certain things are normal and fine, and they're actually not normal. It's just that we've become kind of desensitized to them thinking you know, this is what happens. For example, like the journal model, the academic journal model, and, the gatekeeping that happens there and the publisher perish paradigm. And it's like, well, actually, before the 40s, like most research was blue sky funded. And it was like you were funded as a researcher because let's say the government or whatever funding body believed in your brain to do cool things. And so you like, here's this money. You don't have to have a project driven funding model. It's not necessary, but yet it's become the status quo in the same way that like junk food and processed food has become a status quo for food, but maybe food isn't meant to be cheap and accessible. Maybe it's supposed to be a little more scarce and nutrient dense, so you don't need to eat as much, and maybe it's supposed to be a little harder to work for. Like I think we've just been so accustomed to thinking food's supposed to be cheap that, we're supposed to get our water in plastic bottles, that we're supposed to, have this academic funding model that is gatekeeping scientists from publishing. Like I like to always tell people in my research, if I wanted to publish something as an independent scientist, it would cost me $12,000 to put it in the journal Nature because I don't have this grant funding to prop me up.
Yeah, yeah, and of course nowadays the cost of publication is zero. That's all prestige and
Yeah.
censorship that's going on there. It's funny. Some of the reactions I've seen online about this is the academics seem to have the least understanding of economics on this. So they tend to act as if when this happens, there'll be no primary research, and so the economy will come to a crawl. So they have this belief that only the academic sector does this primary research, that like the private sector doesn't do it. They just come along and sweep up the goods and go monetize it. And it's very bizarre perception, that they must not understand, like, that, the size of private investment into research is like 10 times larger than the NIH, right? It's like 47 billion or something NIH, and you go look at what the private healthcare and biotech space does, and it's 10 times that. And yet they sort of insist that the only way that we'll ever get these things that have no economic worth studied, which you should stop and say, why are you studying them? They're like, we need people to do this primary research that has, we don't know where it's going to go. It has it has no profit signal, so someone has to do it. And I'm like, well, why? Why are they doing it? If you don't think there's any market reason to do it, you shouldn't be doing it. And then they'll turn around and be like, these private companies, they wouldn't survive if there weren't NIH grants. And I've been through NIH grants, and I can tell you that you can't get one without putting money down first. Like you have to submit a grant application that could probably take you several FTEs to write the grant, several FTEs to create the preliminary research, and almost every grant people submit, 80% of it's done on submission. And you're just asking the government to take it over into the end zone, if you will. So private sectors is funding the application of a lot of those grants. And if it's not the private sector, maybe it's another PI who's working money from another grant that happens to overlap with a field, and he gets preliminary data he can submit for another grant. So there's always somebody funding the submission of the grants. The grants aren't like, they're not throwing money from helicopters at cool ideas. You have to actually commit a significant amount of pre-investment in order for you to apply for one. So this concept of like market failure, that, oh, the market is failing, therefore NIH needs to fund, Tony Fauci ripping out the larynxes of dogs and putting fleas on their heads, or NASA has to be funded so we can get Tang. Like, no, that's not how research should work. You don't build a space shuttle so you can get Tang. You build Tang because maybe athletes want it or something. They're lost in this trap that if you don't have this primary research being funded by government, then greedy capitalists will distort science into a way that it'll become corrupt. And they can say this. They say this with a straight face after we just witnessed what happened with the WIV, the Wuhan Institute of Virology. Tony, like, he's out there like funding the gain of function research and then hiding the fact that he did it, and then coming out with a vaccine that they profit from. And it's mind-boggling that they can't see that, no, there isn't any accountability in government funding, and that's the problem. In the market, yeah, companies can do nasty things, but people usually get fired, or their stock goes down, or the market has many more mechanisms to hold them accountable when they go off the rails. It doesn't always work, but putting unaccountable people in there doesn't make it better. That's what I think the spastic emotions we're seeing coming out of the academic institutions right now is that it's the end of science because we might actually penny pinch at the NIH.
Yeah, meanwhile, the most corruption and distortion has happened due to this government funding model, this grant funding model that's been basically propping up labs that are doing research that fit within their projects, guidelines. And then that's preventing labs from maybe doing things that they're interested in actually just intuitively, or they think are important that maybe they can't get funding on. And it's pigeonholing science, especially translational medicine and that kind of science, into a certain direction that is wholly unhelpful in a lot of ways. And I mean, if we look at also the reproducibility issues, there was I think a paper that came out of, I don't remember what lab it was. I think it was somebody in California, and they did like an
Probably
analysis. What was it?
Johnny Needis does a lot of work on that front. It's probably
Yes,
him.
I believe it was him, and I think he found something like, it was like almost 80% of papers can't be reproduced or something like this. It was a crazy amount. And it just points to, I mean, there's a lot of issues there. It could be like, unknown unknowns, poor controls, things like this, but it could also be like, maybe the projects we're working on don't matter in the way that we think they matter, especially within the context of the NIH, CDC, that are developing these project guidelines for like developing cancer treatments that like kill the cancer just fast enough to not kill the patient. And it's like we're focused on these symptom management project, let's say, or specific drug target projects at the expense of actual human health and creating health. And so I
Yeah.
think that's where the decentralized model really comes in and can kind of shine.
I learned this the hard way when we had we had several government grants, but one of the largest one was a $27 million grant from NIH to be a genome center of theirs. And we got the grant because we had low overhead. That was it wasn't any like brilliance on our part. What we did is we moved the sequencing facilities out of Cambridge, where the square footage was extraordinarily expensive, and we put it in the back of a shopping mall in Beverly, Massachusetts. So we cut every corner we could on costs so that our overhead was like 20%, and the other people were getting 60% taken from the universities. So the NIH loved that cost saving strategy. Francis funded us, Craig Venter, Eric Lander, um, Bob Watterson at WashU and Baylor. So there was 5 of these genome centers, and we got really good at sequencing one type of plasmid DNA, phosmids and plasmids in general, so that we had probably half the cost of everybody. And that's what kept us in that game. We then ended up building this DNA sequencer that we were building the side with private money. We had applied for the $7 million grant. We got the grant when it was about 80, it was 80% done probably on completion. But then that grant had spelled out a 3-year research program of all these other tools we're going to build onto the sequencer. And by having that large grant, we were somewhat obligated to 2 masters. We had to keep going after those research goals, and even as the market was telling us we don't want those tools, we were still putting money into them because hey, it was dilution free capital. Our shareholders are getting a free ride on that. That's great. It has good prestige. But it ended up putting our company in a direction of putting too much investment into something known as mate pair sequencing, when the market was going to paired end sequencing, which is uses less DNA and was getting more traction. Illumina was our main competitor, and Illumina had less government funding. They were more privately funded, and as a result, they were more responsive to what the market was demanding, and they doubled down on paired end reads, and they beat us in that domain. While we were still kind of capitulating, being like, well, the market's telling us to go this way, but we have this grant that's funding all these people to go this direction, and maybe in going this direction that the government's telling us, we'll discover something that will get us to this other end goal. So I think the lesson there was that the frequency of study review on your grants isn't as fast as the market review. We did apply at one point to change direction for we were building a sequencing chemistry using polymerases. We applied to NHGRI saying, look, we want to switch to using ligases. We're getting more luck over there. That took us about 6 months to a year to get them to bless that. In the private sector, that would have taken a week. We would have been like, look, the data's here. We know we have to go left. Switch now, and make the call because we don't want to have to wait for approval from an outside body to tell us that we're drilling in the wrong place. So there's the feedback mechanism from the market and from customers is you can't sever that. Otherwise, you're investing money in places that people don't find value. And if you don't find value in the marketplace, you don't have a perpetual margin machine that's going to help continue your R&D. So it always has to be directed by people willing to give you continual cash for what you're doing. And the grant cycle is just too slow. And the other thing about the grant cycle is the grant cycle attempts to be the market by bringing in a sub-selection of the market in the study section. So they might bring in 12 experts. Like these are 12 gurus who would come from the market. They're going to review your grant and say yes or no. But that's always a subsampling of the intelligence in the marketplace. And they also often have conflicts. They have their own companies they're usually involved in. They're not just people that are they're not like academic monks in white cloth. They actually all are brilliant and tied to many industries. So there's some conflicts that you have to try and navigate. But the better thing is to actually get feedback from everybody in the marketplace than from 12 select people that are in the study section. So everything the NIH is doing is creating a statistical sampling problem. They're going in and selecting a subset of intellects that say, this is where we think the research should go. When if you're in the market, you're listening to maybe 30 customers a month telling you that, no, we want you to build X, Y, or Z. And that's what's most valuable to us. So it's set up in a system that's meant to just distribute money from the money machine with arguably with some sort of blessing from sages. Some oracles come in and say this is the right way to spend the money, but it's never going to be an oracle that is like a poly market type of oracle. It's never going to be what are those betting markets they have now in crypto, right? You just don't have enough. You don't have enough voices of customer, if you will, when you're going through that model. So. I think that's where we learned that the market, the free market mechanism is as flawed as people may think it is. We don't have a better system than that. And building up all these centralized means of distributing fiat money are always going to underperform that.
I think it would be interesting, well, while we're on the topic, you mentioned fiat, you mentioned Bitcoin briefly. Maybe we can talk about the stark differences between those two forms of money as it relates to how science is conducted, and you've kind of alluded to it. But then also maybe mention a little bit about how you got interested in Bitcoin, the blockchain, and also finance in general, because it's clear that you're very like well versed in these areas that a lot of scientists are not. So was that just by default as you're trying to navigate the space, or
Yeah, so when you end up stepping out of academia, and you have to start companies. You get a real rude awakening as to how to raise money and expectations on return of investment, and it sharpens your focus onto projects that are going to deliver as quickly as possible on return on investment. And you learn about economics and time value of money. Where Bitcoin came into my life, because it was obvious. I started these companies in 2000. It wasn't around then, but one of the companies I started after we built a sequencer that went to market and was used in a lot of cancer programs. I started getting asked about cannabinoids from people, because these are non-toxic drugs that are being used by patients in cancer therapy quite effectively, and there's no support for them. Like the NIH model just didn't entertain that, because there. I think the reason it doesn't entertain that is because there is limited pharmaceutical interest in nutraceuticals, because they can't get patents. They need patents to get through the FDA, because the FDA process is so damn grueling. They will never approach it with something that's a generic. And we can talk about why the whole patent system is probably retrograde as well. But the FDA is even more of a retrograde than the patent system, in that it creates such a massive hurdle that the only way you get through it is with bribery and wrapped up as PDUFA fees. So the pharmaceutical industry is not interested in anything that is cheap and anything that they can get that isn't exclusive. So the whole cannabinoid space wasn't being addressed. So I started a company to go and sequence that genome, put it public, and hopefully bring that market a little bit more mainstream, if you will. That if usually what happens if you sequence genomes and put them public, that the science in that field accelerates because people can now do knockouts. They can now do genetic manipulation of the plants. They can learn about the biochemistry of the plants so that they can figure out what to feed it to make it grow faster. Right? There's just a revolution occurs once you know what the genetics are behind a particular organism, and if it's public, it lets a thousand flowers bloom. No pun intended, but so, we did that, but in doing that, I realized that this is a horrible idea from a banking standpoint, because that entire industry is unbanked. You want to try and get funding on kooky ideas in the research space. Don't go into the cannabis space, because it's nearly impossible to get funding unless it's private funding. But then that private funding probably has to be from outside the United States, or you got to move to Canada. When we started in 2011, Canada wasn't even legal. And when Canada legalized, many of the people I knew in my field in the cannabis space just moved. They picked up and went to Canada, because they're like, it's never going to be banking free here in the United States, and it still has all types of banking restrictions on it. So that opened my eye up to Bitcoin, and when I saw how that system operated, I realized this is a ledger. If you can track money, you could do peer review on chain. Right? You don't need these journals anymore. The journals. As you mentioned, this $12,000 fee to publish is ridiculous, right? That's considering how easy it is to publish on Nostr or online anywhere. Um, they don't really bring anything to the table. They bring a network of people who could potentially review your paper, and that's it. And that network you can now reassemble online through Twitter, through LinkedIn, wherever. There are all these networks for scientists that you can use to sort of collect people to review your work. The only issue is there's no pricing signal in the peer review system. Right now, they have it set up so the journal takes $12K. They give nothing to the reviewers. The reviewers are expected to do the work for free in a weekend. They usually hand it to their grad students because they're too busy. And oftentimes it's the only people that will pick up the free work of peer review are your competitors, and they'll act like, oh, I don't have a conflict. I'm not really their competitor. They'll review your paper, they'll trash it so that they can learn from it and then get ahead of you and push you back. And the editors try to police all this very poorly, because they don't want to ruffle too many feathers, because their job is to keep all these people networked so that they can find reviewers. And then once the papers get out, they start charging the universities to buy them back again. You assign your copyright of your work to the journal, which is crazy. They then have it and they can sell it to universities or to individuals for $50 a download. And then they have page spaces on their either in their journal or on their website where they sell to pharmaceutical advertisers. And about a third or more of the revenue is coming from pharmaceutical advertisement, just like the mainstream media. So it's no wonder they've become a gatekeeper because they're not going to publish something that tarnishes their number one paycheck from Pfizer. And that alone makes them no more honest of a purveyor of information than CNN. They like to pretend they are, but look at the pandemic and see that the Lancet went through there, killed people. The Lancet killed people with a hydroxychloroquine study. There's no doubt about that. They're complicit in murder, and they should be brought into a RICO case. I hope RFK does that. Nature has arguably killed people because they publish and still stand behind the proximal origins paper, which is complete fraud, claiming that this virus didn't come from a laboratory. And as a result, there's no gain of function change in regulation in 4 years, while H5N1's coming out of another lab, probably in Georgia. So they're involved in a racket of distorting public information about what's actually happening with these pandemics and all this culling of birds and stuff. It's grotesque. So that whole system needs to be, wiped out and annihilated and rebuilt with, I don't expect people to regulate them into success. What's going to have to happen is like the Bitcoin community is going to have to just build a better system and make them completely obsolete. And they can do that by disintermediating the journal entirely. You can just do peer-to-peer review. Just put bounties up online that are paid in crypto and $500 to review up front, 500 later, whatever. Pick your own economic model for this. As long as it's transparent and people know that there's money moving, the reviewers are getting paid. Yes, that has to happen. And you can call that bribing reviewers. But right now, you're paying the journal and expecting the reviewers to donate their time for free and for it to work out. It's never going to work out. You're always going to get people. The only people who jump into that system are people who want to throw sand in the gears of the publication. And they have no motivation to do this quickly. The moment you put pricing signals into peer review, you can create a market so that papers that need to get through quickly can get funded with twice the amount, 10 times the amount of money. Small papers that don't have a tremendous amount of content in them can go maybe through a fast 1 week review with one or two reviewers. And you can put bounties on making them go fast or go slow like you can on the way you ship products from Amazon. That needs to happen in order for there to be any veracity in the quality of the work. The moment you rip pricing signals out of a marketplace, you get the U.S. healthcare system or the Canadian healthcare system. There's a good book from John Goodman called Priceless, which discusses this. When you pull price signals out of markets, how they decay, the directions they decay, and what things fall apart. But we don't have any price signals in what we consider to be the most foundational aspect of science, which is peer review. It is communistic. It's a complete disaster right at the base layer of science. So I'm hopeful that this resonates with the Bitcoin community because they believe in consensus. They believe in building truth sets. And many of them were abused by the pandemic, because we have this fiat system over here creating false science that's now affecting their civil liberties and their health. And no amount of Bitcoin can buy back time. So I'm hoping some of them get incentivized to venture into this space and help us rebuild it.
Yeah, and I think also if you're adding in addition to the financial incentives for reviewers, and also adding in like a non-anonymous merit-based system where it's like as you do reviews and those papers are successful and the results hold up, you get like a little gold star next to your name as a reviewer so that you could then be chosen for other reviews, and then there's like competition happening.
Hey, it's a good analogy is like the taxi cab medallions versus Uber. The Uber drivers and the users of Uber get rated. You throw open an Uber car, you're not going to be easily allowed in another one, right? Likewise, the drivers, if they're poor drivers and don't treat you well, they'll get a rating. I think reviews can happen the same way, and this might solve some of the PhD problems, right? Like we've got a lot of these students who are starving. Well, they could be making money on weekends reviewing papers while they're improving their domain expertise in the process. What's wrong with that? Why would academics get in the way of that? At the same time, the universities are turning around and allowing their athletes to get monetized with these new athlete words, which I'm all supportive of, but they won't allow the same thing to occur with their PhDs. The PhDs there have every right to earn an alternative living to supplement their income if they need to, and if it's working weekends reviewing papers, why are they forcing them to do that for free?
Totally. And I think maybe an example of like the centralized versus decentralized way of doing this is like community notes versus fact checking.
Yeah.
It's kind of a similar situation. I could even foresee, like, in a future decentralized journal model, like, it could be a community notes type situation
Well,
in addition to the peer review where it's like you're getting feedback from multiple voices with different perspectives. I think it's
yeah.
valuable.
Some of it might evolve on Substack. I mean, I know Substack isn't only science. There's a lot of other content on there, but a few of us are publishing stuff on Substack, and we're not publishing it with any intent of it being a perfect replacement for peer review. I usually put things up there that are less mature than that. That might be like at a poster level, right? Where it's some experiments you did in the last few months. You want to share it and get feedback. It's probably not ready for what you would consider full peer review. Although the only reason people pile up science before they submit it is that the review process costs 12 grand. So when that happens, you're not going to submit like a month's worth of experiment into a 6 month peer review process that costs 12 grand. The fact that it is that expensive and that long means that the cycle time of science gets slowed down. People basically bin their science into bigger buckets before they publish now because of that hurdle. If you remove that hurdle or put a market signal on that hurdle, people might publish every month. And they might do it on something like Substack. And I've already had several of the Substacks that I've written be cited in other peer review papers now.
Awesome.
Um, from the DNA contamination front, I had one last week. I was just playing around looking at this hop latent viroid that's devastating the cannabis field, and I was sort of publishing my stuff as a real time journal. And one of the premier plant pathologists in the space picked up on it and published a paper replicating some of that work in a peer reviewed journal in Frontiers. And I was like, all right, excellent.
That's
He didn't have to
awesome.
do that. Like in most cases, academics, they won't cite it if it's not like at least in a preprint, right? And they'll just skip over it. But this happened to be a very honorable person who said, "Oh yeah, that's a really good idea. I'm going to replicate that, include that in my work, and see if it pans out." And so he had, and there's a Substack citation inside of a Frontiers article. So we have some of the tools to do this now. The real question is whether Substack is resistant. Like they could get acquired and then turn into Facebook. You just don't know if they're not decentralized enough to maybe resist a state level attack like that. But Noster comes to mind, right? There's a couple of these other platforms that have publishing capabilities that could decentralize the sort of CEO founder risk that we have at Substack. But at the bare minimum, we have easy tools for researchers to build their sort of online journals, if they will, or notebooks
Hmm.
that those are being shared at a faster rate. Yeah, there's going to be slightly higher error rate because you're seeing people's early results, but you also get the journal of negative results with it,
Yes. So
which
important.
doesn't happen right now.
Exactly. Yeah, and so many people are spending money and time repeating experiments that were already done and shown with negative results, but that just wasn't published. And so I think that's absolutely super important as well. And you were talking about the blockchain briefly. So what does it actually look like when you're like logging your results on a blockchain and timestamping it for IP reasons or what have you? How does that actually look in like a practical sense?
So we did one project where we sequenced the cannabis genome in 2018 with PacBio. And Specific Biosciences has this really cool single molecule sequencer, and the cannabis genome up until then was only sequenced with Illumina sequencing, which meant it was really fragmented in like 100,000 pieces. So we couldn't really organize chromosomes from it. And PacBio has this thing that does like 20,000 base pair reads, and we were able to build it into like a chromosomal map and a really beautiful picture. So that was a project where the Dash blockchain funded it. Dash is a fork of Bitcoin, and they put in sort of a federated model where the miners of that network get to choose how to spend 10% of the block rewards. So, in Bitcoin, the block rewards all go to the miners. In this case, they call them nodes, where if they host a copy of the blockchain and provide some services for the network, that they get to then vote on how to spend 10% of the rewards. And it's a bit of a sort of a pooled model, if you will. And so they vote on these projects that they think may help the success of the network. And in one case, they liked what we were doing is they like, yeah, let's fund the sequencing of this genome and this model of people using blockchains to record the scientific record. Now, at that time, they didn't have a Dash drive like where you could put large data sets, and blockchains aren't good for putting large scientific stores of data. They're only good for really recording digital timestamps of when data occurred. And you can do that with a really quick hash function. But the data itself, you need to point to somewhere else, like IPFS or maybe Mega or some or BitTorrent or something. We used IPFS for that project. So we took the big sequencing data, threw it in IPFS, and then every time we had an experiment we thought was meaningful, we would hash that data and spend it into a transaction on the Dash blockchain. And that would record it on a chain that was immutable, so that you could always prove when certain data was generated. Um, and then when the project was complete, so it was funded in July of 2018, and we finished it in August, and put it public in August. So from funding until publication was like 3 months. We then put out a bounty for reviewers, three reviewers, $500 in Dash up front, 500 on completion of the review. Review's got to be done in 7 days. They all got paid out, but they all put their resume public and the review public, and the fact that they received half and half of the funding, so everyone's transparent on what the incentives were. And we got a great review back. There were critiques. It wasn't. I wouldn't say you deem it. It was a perfect story. One person brought up that we could have analyzed the genome a couple different ways, and they're right. We ended up redoing it. And the other two people more or less signed off on it, saying, "Yeah, this is a significant achievement in compared to where the current references were." So, in a matter of like 5 months, we did what would have taken 2 years, and it was all the whole genome cost about $50K, and so we got it sequenced, published, and reviewed faster than an entire review cycle could happen in a traditional project. So it's very quick funding, very effective process for this. So the blockchains are really just being used as a time stamping mechanism and as a mechanism to coordinate peer review. Our peer review is coordinated internationally, so it was easy for us to get people crypto 24/7. We didn't have to wait for a bank to open on a Monday morning to try and wire money to another country. And we didn't have to worry about PayPal interceding if we decided to use a reviewer who was canceled or controversial. So this all could now, maybe Dash isn't the right chain to do it now because they're not as the chain isn't as heavily used as Bitcoin, but it is there as an option with has a faster block cycle, and it has cheaper transactions. And now Bitcoin has Lightning and a couple other functions on it that maybe some of this can get done there. But the architecture of the chain isn't terribly important as long as you believe it's immutable. And it's going to have longevity to it. Like I think Bitcoin is obviously going to have the most longevity, the most proven history. It's got the most hash rates. That's probably the most secure place to put really important studies. But all studies don't necessarily need to be on the base layer of Bitcoin taking up space. For those not familiar with Bitcoin, there is this function in Bitcoin where you can print into the op return it's like a field, if you will, a notes field. And that's what people are using to stuff hashes. It's called a hash and stuff mechanism. You hash your file, it creates a unique fingerprint of your file. You spend it into a transaction in there, and that gets distributed to all the nodes and put into a transaction, and it's globally forever in the blockchain, ever forward. And the actual large data sets, you gotta link somewhere else, and the websites that contain that link are a weakness. Like we had it on our website, and our website has been hacked and attacked by people since we did some of this stuff. But you could put those links on Nostr, which are more decentralized than our website. So the connection between the timestamp and where the data sits should be on something else that's probably more secure than your own web server like Nostr.
Got it. And so for that fingerprint, the file fingerprint, if you were to make any edits on that file, would the fingerprint change so that there's a way
Yes.
to prevent? Okay, got it.
Yeah, it is. That's an important aspect. So the way these SHA-256 hashes work is if you change one letter in that file, the whole thing changes. The
Got
whole
it.
fingerprint that comes out changes. So you can tell if it's been tampered with. You can't go the other direction though. You're unidirectional. So you can't take a fingerprint and figure out what the original file was. So anyone seeing the fingerprint of the blockchain can't know that came from this cannabis genome. All right. So you have to have some other site that says, here's our sequence data, here's proof of the timestamp. And if you run SHA-256 in this file, it's much like an MD5 hash sum you get when you're downloading data to ensure that the data you downloaded off the website matches the hash from the manufacturer. That tells you a virus hasn't inserted itself into the software, and you have the exact thing that was presented. If there's a change, all you can know is it's been tampered with, but you don't know how.
Mhm.
The unidirectional nature of that's actually kind of important for Bitcoin, but we're leveraging that just to say that this file creates this unique hash. Nothing else in the world can create this hash because the hashes are long enough in length that there's great videos on how unique these numbers are, but they're enormous numbers. They're like 2 to the 256 power in terms of their size, which is like, imagine all the sand grains in the universe, right? It's that's sort of a number. No one can guess that file, no other that hash. No one else is going to create a file that collides with that hash because it's too big of a number space. So those unique fingerprints are really important. You change one letter in your file, and the whole thing changes.
Great. That's very good to know. So let's pivot a little bit into science. I asked people if they had questions for you, and I definitely had quite a few people asking about both the jabs and cannabis and cannabinoids and their use in
Yeah.
medicine. So maybe we can start either direction. Which one makes the most sense for you?
Well, I think the jabs are important to hit on just because I don't know that they're going to. I just read this morning from Mary Talley Bowden that like another 9 million kids have gotten jabs since the election.
Oh my god.
So it's like yes, they are. They are, less people are taking them, but the people that are taking them are the kids because they're getting forced to.
Yeah.
And for whether school systems, vaccine schedules, what have you. So I agree with Jack on this front that we can keep pounding our table on people to pull these, but we have to start focusing on how to fix the people who are going to hit by them because too many have been hit by them, and we can't reverse that. So it's great to stop them going forward, but therapies need to start hitting the forefront of the discussion. So he and you have been, I think, doing great work highlighting red light therapy and all of these other frowned upon fields of medicine that are cheap and effective and considered voodoo, if you will, by the mainstream funded fiat machine. I've been focusing on these cannabinoids because I feel very similar about them. Is that they've been demonized because they are a massive threat to the pharmaceutical industry. When you start looking at the areas that they touch on the pain market, which is like an 80 billion a year market. They touch on epilepsy, they touch on autism, they touch on potentially vaccine injury, they touch on you go down the list, and this is a much bigger market than the entire vaccine market that they can have an impact on. A lot of states that legalize them show there's like a 10% decrease in alcohol consumption. So it's another whole, multi-billion-dollar industry that is petrified of these things being legalized. So as a result, there's an entire institution at the NIH called the National Institute of Drug Abuse that doesn't look for drug benefits. They only look for abuse. So they
Hmm.
litter the scientific record of all the problems with cannabinoids. If I could only have such an institution, right? I won't be surprised if one gets built. The horse-based institution will probably be built shortly. But as you can imagine, that skews a lot of the scientific record in the direction of like cannabinoids causing schizophrenia. And I saw a paper the other day that was trying to blame some guy who chopped his ears off and cut his dick off for after smoking weed and taking kratom. It was-
Oh my god.
yeah, I mean the papers that come out are just like, oh, gee, you didn't bother to highlight the fact the guy was taking meth. I mean,
It's still reefer
it was
madness, right? It's like
it's
literally.
so reefer madness. Not to say that they're obviously every drug can have negative effects, and these aren't panaceas. But it's very difficult to sort your way through the literature if you're starting from ground zero. When someone says, "Hey, these cannabinoids might help with your kid's epilepsy," a mother is going to be absolutely like petrified trying to dig through. 9 out of the 10 papers out there are going to talk about it causing you paranoia and cutting your ears off. And the real heart of the matter is a much different story there. So they're very non-toxic. If you look at the LD50 on drugs, that's the important thing to look at is a lethal dose versus a therapeutic dose. And there's a huge margin of error with these things that you can't find with Tylenol. You can't find with a lot of the over-the-counter drugs that are out there. Um, Tylenol is its own rabbit hole. It's actually a cannabinoid mimetic that no one will tell you about, but it makes a toxic liver byproduct called NAPQ1. It's a prodrug. It turns into AM404, which is not a web browser error. It's actually a drug from Andreas, what's his name, McKernan out of Boston. But that's a cannabinoid mimetic that they're trying to give you with Tylenol. But it comes with a toxic liver byproduct. If you take too much of it, you kill yourself. The cannabinoids have the feature that you want without the liver byproduct issue, but no one will tell you that like CBD is probably a better drug than Tylenol for most of these situations. So there's all of this stuff going on in the pharma industry that demonizes that thing, and as a result, people have had to vote this thing over the counter, like Mary's trying to get done with ivermectin,
Mhm.
state by state, and the red states are fighting back. And there's a big mess in how it's getting legalized. They've legalized it in two different flavors, like in a hemp flavor and versus a cannabis flavor, and the hemp flavor is banked, and the cannabis flavor is not banked. And in the hemp market, I'd advise people to stay the hell away from the hemp market right now. It is just contaminated with spice, K2, all types of drugs are getting sprayed on hemp and sold at gas stations,
Mhm.
including kratom, including opiates. Like it's gone totally off the rails, which is upsetting because 5 or 10 years ago, the people who are delivering CBD oils to kids with epilepsy, that was the real Dallas Buyers Club many years ago, and it saved a lot of kids' lives. But now these hempsters have come in there and used that loophole to sell all types of drugs sprayed onto hemp into gas stations and smoke shops and what have you. But anyway, so I got involved in this whole cannabinoid field because it looked like a way to decentralize medicine. Can we get drugs that you can grow in your backyard and get a better industry of information around them so that we can breed them more effectively, we can test them more effectively than the FDA is ever going to test them, and we can give people confidence that there's safe. You can grow safe drugs in your backyard. Um, how that intersected with your initial question, I've lost track of. I think it was how it intersected with Bitcoin, I suppose.
Oh, well, yeah, I mean, I'm really interested in talking about the cannabis and the cannabinoid story and the jab story, but I mean, just to chime in
Oh,
briefly, so I think there's so many different types of cannabinoids, right? I feel like new ones are being discovered all the time in different plants outside of cannabis as well, and of course, the body makes its own endocannabinoids as well, and actually, the lab I did my postdoc in at Penn, right when I was joining, they were publishing a paper, I think it ended up in maybe Cell or Nature, and they were basically showing that there's a specific group of microbes within the gut that facilitate the production of a lipid amide that's basically serving as an endocannabinoid. And if you
have
have
you?
this group of
Is it
bugs,
is it PEA by chance?
it was oleamide.
Oh, good. Yeah, excellent. Yeah, that's a very
Yep.
important one.
Yep.
Yep.
So they basically found if you had, I think it was a group of bifidobacteria, actually, a specific subset of bifidobacteria in the gut.
Oh, I gotta ask Sabine about that. Yeah, great.
Yeah, so this was in rodents, but I think they're moving on to to human studies. But anyway, they showed that if you had these bugs making a lamide, and then you have these animals exercising, they're more likely to self-select exercising in the future because apparently this endocannabinoid can help to trigger the dopamine response to exercise, and you have that reward mechanism in place, and the animals that were depleted of this subset of bacteria didn't self-select exercise in the future, and because they didn't have this dopamine reward, and then they provided the alamide back as a supplement, and it restored the dopamine response. So it's like there are really interesting things going on inside of our bodies naturally from the bacteria or from our own cells that are involving these pathways as well. And so I think it's a really interesting interface of like what nature can provide and what our own bodies can synthesize to create different benefits for the system as a whole.
It's such an important point because they've that entire ECS, the endocannabinoid system, has been horribly suppressed. For reference, one of the most common G protein coupled receptors in the human body is the CB1 receptor. All right, so this is one of the most common. 25% of all drugs hit like G protein coupled receptors, and the most common one in the human body is illegal.
Mm,
Right? It gives you a sense of like how much they've distorted what's taught in medical school. Right? There's very few. Today, there's a few, but maybe 10 years ago, almost no medical schools taught about the endocannabinoid system. Like doctors weren't even aware that this system existed, that we had endogenous compounds that hit it, and that
mhm.
now you're bringing in microbiomes. It's incredibly an important topic. But yeah, there are a lot of them, and a lot of people tend to think of cannabinoids as just THC, which is a big that's success of the media trying to scare you about cannabis. That this one compound in there happens to be psychoactive, but there's 100 other cannabinoids that aren't. You've probably heard of CBD. Many people have probably taken CBD, and CBD, great, that's out, has a lot of functionality to it. But what's been in the pipeline, in fact, if anyone comes to CanMed, you'll see what's in sort of the cannabinoid pipeline. There are all the acid forms of the cannabinoids, which aren't psychoactive. Even THCA isn't psychoactive until you heat it. And when you put that acid group on there, it changes the bioavailability of the compounds. Oftentimes, they are more bioavailable when there's the acidic form there. Sometimes also changes the receptor profiles that they hit. After CBD, people started playing around with CBG, which is cannabigerol. That's the precursor to both CBD and THC. The plant makes CBG first, and then it gets folded into either THC or CBD. A third compound it can be folded into is cannabichromene. And not as much is known about cannabichromene. It's being used. It has some similar properties of CBG. It's an anxiolytic. It's helpful for sleep. It fights against MRSA. It has some good properties for acne. You can probably hit like ChatGPT is pretty good at spelling out a lot of these different differences between the cannabinoids. We're now finding all of these other cannabinoids that have a longer or shorter carbon tail on them. When they're shorter, they're less psychoactive. When they're longer, like there's a THCV, which has got only 3 carbons on the side chain, and that one is known to suppress appetite. So there's some papers that came out recently showing like a 10% drop in BMI when the people went on THCV for 90 days. So here's something that could tackle Ozempic, right?
Mm-hmm.
Maybe it's not dropping people's weight as much as Ozempic, but again, it's not coming with bone loss and blindness and all these other issues. If you add 2 carbons to that tail on THC, it turns into THCP, which is like 30 times more psychoactive.
Mm-hmm.
And that's what's being found in a lot of these hemp stores. They're spraying. The plant makes. There's only been a couple cultivars where they can find sub percentages of that made. So they're synthesizing it instead and spraying it onto hemp and claiming it was made by the plant to try and get it through over state lines. And the DEA hasn't really. Not that I'm a big fan of the DEA hyper-regulating this, but the patients and the consumers don't know that they're consuming something that's 30 times more potent. I mean, that can get you in trouble. And so it's really more of a fraud issue that they're supplying these compounds with synthesized in a lab, and it's not well characterized as to whether the synthesis is pure, whether there's any background side products in the reactions, all this stuff. Um, so the varins are the ones that have shorter tails. So CBDV and THCV are both exciting cannabinoids that have come out because they do different things than CBD and THC themselves. I think CBDV is being looked at in epilepsy. It looks like it has a better profile than just CBD alone. So anyway, that's probably like a dozen of them they're actively being studied today that we know something about. There's been a bit of a focus on those carbon compounds, 21 carbons in these cannabinoids, because they're lipid soluble and they get through the blood-brain barrier. And you get different biochemistry when you're dealing with lipid soluble drugs than water soluble drugs. There's a host of other compounds in the plant, which are known as the flavins, the flavonoids, the canflavin 1 and 2 being looked at in cancer. These are more water soluble, and the kynurenic acid, which they're finding in the leaves. So it's a bouquet, if you will, of it's a pharmaceutical, factory, if you will, of compounds that have had they've been well studied, used in the human population for thousands of years. So the ADME tox side of going down this pathway is that's the number one thing that will drop you in a lot of clinical trials is you can't get through the ADME tox side of it. But we have good confidence with the ADME tox that's in cannabis because humans have been using it and consuming it for so many damn years that it's unlikely any one of these compounds is lethal.
Mm-hmm.
It's kind of gone through that selective process, the breeding process already, where we've somewhat cohabitated with this thing, and that field will quickly bring you into other plants. It will bring you into fungi like psilocybin. We do a lot of work with psilocybin as well, sequencing the genomes of those of psilocybe cubensis to find out the pathways that make the entourage that's in those. It's not just about psilocybin. They make 4 or 5 other compounds as well, tryptamines that have different properties than psilocybin. So each one of these genomes you can crack. You can begin to look at the pathways that make the compounds and all the side products they make, and usually you find that all the side products are just as therapeutic as the one that's known for its recreational use. They just have slightly different affinities for different serotonin receptors or what have you. So I think there's a lot we can learn by sequencing medicinal genomes and putting the information public so that it can pull the entire sort of nutraceutical market forward, if you will, without having this lording control at the FDA over what drugs make it to market.
Yeah, I love that. And I love that you mentioned like the entourage and their entourage effect. And it's something I think about also in the context of food. And Jack always talks about how food is a barcode of the light environment it was grown in. And depending on that light environment, you're getting different micro and macronutrient ratios. minerals, cofactors, everything like this that are all coming together to create something that provides both energy and information to the body so that the body knows what to do with it. And I think in the context of like cannabis, for example, anybody who's taken like a THC isolate, like an edible, for example, or even just, some sort of a THC isolate versus a full spectrum with terpenes and all the good stuff in there, the effects feel quite different. And even like terpenes by themselves. So like I've taken limonene by itself is also quite energizing and like stimulating in like a very positive way. And so I think the terpenes are really interesting, and also thinking about the light biology here. And it's something I want to look into in the lab, but looking at like the absorption and emission spectra of these different molecules. And that has, of course, has implications about the growing environment, right? So if you're not growing outdoors, if you're growing indoors and there's no UV light, now these molecules that could have potentially been carrying some UV photons into the body, let's say, maybe don't have access to that because their ring structures didn't ever get, exposed to UV light during the growing conditions. I think it's super interesting.
Yeah, we've totally bastardized this by when they've quote unquote legalized it. Everyone has to grow it indoors. The light structure is totally different now, and that has created a total mess of the microbiomes of the plants as well. Because when you grow indoors, you try to start off like in a sterile environment, like everything's clean, right? But it's not really, because the seed itself has all types of microbes in it, and then the soil you bring in, or the hydro you bring, eventually this boy in a bubble gets infected by something, and it's not like a full-blown natural infection. It's a very niche, one species dominating infection, which creates all types of havoc on indoor grows. So indoor grows actually have, you would think they'd be really clean, but they have a really hard time staying sterile and dealing with many of the microbial populations that they're tested for. So oftentimes outdoor growing ends up being cleaner because you have natural pests out there. You'll hear of indoor grows getting like aphid infestations, and like
Yeah.
that just happen outside because there's ladybugs, right? And they're predators around. So the really good growers have to have these like IPM programs in place where they try to recapitulate as much of the outdoor environment as they can inside, without getting pesticides and all the other things that they get tested for in the mix.
Hmm.
But even the pesticides are hard for them to deal with because they've made the mistake in some cases of using drywall. And drywall is like this chalk system that absorbs pesticides and it breathes them back out when the humidity fluctuates inside the groves. So it is there's, I like your analogy on the barcode of the light that's in there, because I do think that plays a role in the cannabis, like cannabinoid and terpene profile that these plants are generating. You can take the same cloned genetics where we know it's the identical genetics, and separate the environments and grow them and get different expression. Now,
Hmm.
usually you don't get a dramatic change in maybe the cannabinoid ratios. Like if your plant's going to produce 20 to 1 THC to CBD, or vice versa, it's unlikely you're ever going to get that switch based on environmentals, because there are some really hard genetics to that. But the terpene profiles are very responsive to the environment. Like those do change quite dramatically. In fact, there's some studies showing if you spray plants with antibiotics and wipe out the microbiome, the terpene profile completely shifts. So the terpene profile really is a communication, a chemical communication layer with the microbiome of the plant.
Yeah,
And
I mean, oh, sorry. I was just gonna say it's making me think of an analogy between like identical twins, and you put them in completely different environments. And sure, they're gonna look the same, but there's gonna be a lot different about them over time, especially.
Yeah, yeah, indeed. And that's the exciting thing about working in the cannabis field, and any plant field, is that you can make clones of things, right? It's something that's very easy to do. Doesn't require any sort of genetic triple Lundys to do. You can cut cannabis plants, put them in a little bit of plant hormone, and they'll reroot, and you have genetic clones. There may be some mosaicism in that process that affects it, but they're predominantly, you've kind of taken out that one major variable. Um, but this impact on the people that eat them or consume them, I think is very important. That goes back to that. I think we had a conversation about this offline, that David Sinclair paper that talked about xenohormesis, and how
Hmm.
when you stress plants, they produce a different secondary metabolite profile that's meant to signal to the seed transmitters that they're to prepare for caloric restriction. It tends to dial right into that, the rapamycin pathway. So that, hey, there's the environment shifting. I'm going to produce things that signal to people who are going to spread my seeds to hold on to them tightly, right? Because they may encounter starvation. So that I think is helpful in cancer as well. Like a lot of people are making cannabinoid oils for cancer,
Yep.
And if they're Warburg cancers, you might need a different one than if they're not, because the whole Warburg effect isn't necessarily driving every cancer. But to the extent that it is, it's you want something that's going to zero in on the mitochondria and alter ATP production to try and get that back in line, and that might require a different set of cannabinoids than what you'd use for something that's not believed to be of that etiology.
Yep. And now that you're mentioning that, I mean, there's so many different ways to get cannabinoids into the body. You have vaporizing, you have smoking, you have topicals, you have tinctures. I'm sure there's others as well. And it's making me also think of, I've heard you speak on other podcasts, but it's maybe good to mention it here, that a lot of the pesticide contamination in vapes, for example, and there was that whole thing with vitamin E acetate and
Oh
popcorn,
God,
but
yeah.
you were debunking it. Want to talk about that a second?
Yeah, so before the pandemic, there was this Valley crisis, which was blamed on vitamin E acetate, and so that was the CDC move. So always second guess it. What's Rosetta? I think is her last name Sagrado. I think it is. I have to forward you the name. I sometimes fumble her last name, but she's done some really interesting work looking at the Valley crisis. And if you remember, the Valley crisis happened right before the COVID pandemic, and one of the signatures was ground glass opacities in the lungs, which sounds just like COVID.
Yeah.
Uh, and it also, what she pointed out is, if you look at the epidemiology of it, it followed this Gompertz curve that, if you remember early on in the pandemic, there's many epidemiologists that were pointing out that this is what pandemics look like. They have this sort of pandemic curve described by Gompertz. Um, there's a Nobel laureate, what was Michael's last name, at Stanford, that was Michael Levitt was all over this. And she saw that fit inside the Valley data. So she's immediately going, this has got to be another coronavirus. Maybe it's the precursor of what actually was released, or another, who knows, another coronavirus. And not that it was just a coronavirus, but that the vapes may have been painting the target for those who got really sick. Because there is more inflammation in the lungs with some of these vapes, and it wasn't relegated to just THC. There was nicotine pens that were in the mix as well. And what stood out to us in the cannabis field is that vitamin E acetate's been in vape pens for 10 years. Why did this show up now? Was there a form of vitamin E acetate that was doing this? And other researchers had pointed out, a person to follow on Twitter is Mass Blank, who's done some work in this area, showing that your own body, when it gets going through some type of inflammation, can create its own endogenous vitamin E acetate.
Mm.
And that the CDC trying to pin it on that was not facing that data. The fact that that's not a signature for, yeah, the pens may have it, but you may be reading that in the tissue, and it may be endogenously generated in the process of inflammation.
It's
So
like firemen at the fire. The firemen didn't start the fire. They were just
exactly, yeah, yeah. So there's a lot of reasons to believe there may have been a coronavirus involved in that, and not that, not vaping's off the hook. But there's other people in the lab space. I know a lot of the people who run the cannabis testing labs, and they kept bringing up pesticides. Like, there are so many damn pesticides in these things that are getting used. What invariably happens is the state will put out a list of like 60 pesticides you need to test for, and it's the 61st one that people move on to use. And so it's this game of cat and mouse. I think the last paper I saw had 327 pesticides being screened up in Canada. And the black market had like 92% contamination, and the tested market had like 6% contamination, but there's still some in the tested market. And those things can enrich in the extraction process. When you go and enrich for cannabinoids, you might move the flower from 20% to 60% or 70% in the oil, but some of the pesticides can go from parts per million to parts per thousand. Just the process of extraction selectively purifies those molecules, some of those molecules, more effectively than the cannabinoids. And so, even though there's still a parts per thousand, they can be very potent at those levels. And the one that's probably would be most frightening to a mitochondriac like yourself is myclobutanol. That thing turns into hydrogen cyanide when you heat it. And that's one of those common fungicides used in the cannabis growing community. It's outlawed in many states, so you tend to find it more in the hemp products that they don't make it through state testing, and so the dumping ground becomes the hemp industry. So be careful what you smoke from the hemp side of things, because the pesticide screens come whenever people screen that market. They're loaded with pesticides. And if you go through pesticides, they're mostly neurological agents, right? They're usually things that are meant to like screw up the neurological system of insects, and they claim they have very little effect on humans at those dosages. But once you get involved with vape pens, they're no longer at those dosages. They're jacked up to higher concentrations, and they can cause all types of havoc. Now that's a separate issue that people end up blaming the activity of THC for all this neurological activity because they
Mhm.
say, hey, this person smoked cannabis or a vape pen that was made from cannabis. They never in those studies go, very rarely do they ever chemotype and do a chemical analysis in the pen. They just say, hey, it's a THC pen. So THC must cause schizophrenia, right? And it's like, no, it's probably the pesticides that are coming along that you're smoking at high concentrations, causing hydrogen cyanide that could be causing your schizophrenic break. That's the quality of science that you find in the cannabis field is it's often that simplistic. Like they look at since it's illegal, it's hard for them to do these controlled studies. They'll just say we collected 1,000 cannabis users and we compared them to people who don't use, but they have no idea what the hell they're consuming on the cannabis side to make those correlations. And so
Right.
it just stacks all this yellow journalism against the compounds in that plant.
Yeah, you're making me also think of like Roundup, glyphosate, and the shikimate pathway. So it's one of its main targets, and that's claimed to not impact human cells because of that. But I'm thinking there's more bacteria in the body than human cells, and so if we're taking
Yeah.
something that's affecting them, we're also affecting our human cells indirectly via the microbiome, basically. So I always think it's just so short sighted to think about things in that way.
Yeah, I mean, isn't there good enough evidence now that its impact on mitochondria, or is it still are they still trying to back out of that?
I can never keep up, but I mean, in my mind, I'm gonna stay as far away as possible from it, for sure.
Yeah, I thought Steffi Snapp did some interesting work on that with deuterium and the impact of glyphosate on the mitochondria, but it's I confess to not being totally up to speed on that either. But yeah, it's another pesticide that fortunately we don't have Roundup in cannabis yet. Someone has filed a patent on Roundup Ready cannabis. I don't know
Ah,
the extent that it's actually being used. The industry is I think fairly adverse to that type of activity, but it's not to say that someone won't try and sneak it in there.
right, and those pesticides are they're used in both indoor and outdoor grows, or
They are, yeah, and the indoor ones sometimes are more enriched because they can't get rid of them. They tend to get stuck in the drywall and everything else. So they're usually in the indoor environment. They do a grow, and afterwards they bug bomb it. And that bug bombing is, the plants aren't around when that's happening. They're just trying to just kill any of the bugs that might be creeping around on the walls, but the walls absorb some of those bug bombs and oftentimes breathe them back off when the humidity changes and the plants get in there again. So
mm,
they have to be really careful about using them. Now, there's a list of ones that they're not allowed to use, and some that they can that are deemed less harmful. But the state, at least the state tested cannabis facilities and dispensaries. I mean, check with your state. We have some of this on our website as to what the regs are in each state, but in a lot of states, they are testing for like at least 100 of these things. And whenever studies are done looking at what's in those markets versus what's in the black market, it's always cleaner in the state market, but it's not perfect because the
mhm.
state market isn't heavily policed as you might imagine. And a lot of the users aren't really aware of this problem. They just assume, hey, I got it at a dispensary, it's better than the black market. And so they don't, they may not ask for a certificate of analysis to prove that it's actually been tested for. But it's getting better. You'll see a couple studies from Paige, what is her name? Paige St. John has done some interesting work at the LA Times, and then Tiffany DeVitt did some a really interesting thing. If you want to scare yourself, read hemphoax.com. They'll show you all the crap that they're finding in the hemp market. That's become the dumping ground for the stuff that can't get through the state labs. But really weird economic dynamic going on here is that the the hemp market is ironically banked, and the cannabis industry that does safety testing is unbanked.
Mm,
And so, just the last 3 years, the hemp market went from nothing into being bigger than the cannabis market. So, all of the all of the growth in the cannabis industry in the last 3 years has really gone into this market that is banked but doesn't do any safety testing on stuff. And now we're just starting to get some backlash where kids are getting these like 200 milligram gummies and parents are flipping out over the fact that they bought them at a gas station. And I think I just read that Texas is going to, go full bore prohibition again. So it's, it's a bit of they call it red state hemp because a lot of the red states that haven't legalized cannabis have this problem. And the
right.
reaction isn't to legalize cannabis; it's to actually, double down on prohibition.
Yeah, it's like gonna make the problem worse. I mean, I'm thinking of like the loopholes like Delta 8, for example, or like these slight
Bingo.
modifications you can make to make
Yeah.
something legal in the head shop. And then as soon as that becomes illegal, they find a new one to like, push into the head shop. And it's just this vicious cycle as long as it's illegal.
Yeah, and the crazy thing is the remnants of the drug war keep this going. Like kids in schools are tested for Delta 9.
Yeah.
So they're
That,
incentivized to use, HHC, which is we don't know as much about HHC. Might be can that might be a fine cannabinoid. I don't know, but it's like no one's studying that. And so and if they once they
Hmm.
make HHC legal, they'll move on to something else that the test won't hit. All right, so the prohibition that still exists on Delta 9, which is a really fairly safe compound, we understand it. Yes, you don't want to be, taking chronic amounts of this your entire life, but it can be helpful for replacing alcohol or for pain or for sleep. But every time they prohibit that, they just push people into using these drugs that are like, Jesse Pinkman bathtub gin that was made last week. You don't really
Yep.
know what it is, and it could have a totally different tox profile. So it's funny that people's response to it is that they see that going on in the head shops and they say, "We told you legalization was a bad idea. Like ban it all again."
My God.
Oh,
So backwards.
you're getting the wrong message. It's
Uh,
that you have one area that's banked and one area that's not banked. So all the business is going to flow to the not the banked region, and that bank market is right now incentivized to make compounds that don't hit drug tests.
mhm. I remember once in high school, I had some, like, K2 spice situation from, like, a
Oh,
gas.
you're crazy.
It was like a straight psychedelic. It felt like I was on acid, but, like, the most paranoid trip in my life. It
Sounds
was so
awful.
weird. It was
Yes.
weird. It actually looked like everything was, like, outlined in a black Sharpie, and I was, like, sitting in a forest, like, so paranoid that somebody was gonna find me.
Oh god. I've never done it. But I that's the thing is I prefer psychedelics. I don't have any paranoia, and that
Yes.
I am one of those people where THC, like, it hits that paranoia bone of mine, so,
Me too.
I'm very sensitive to it, and I end up I play with a lot of the other cannabinoids, but, THC, I can only tolerate in very small amounts, but,
Yeah, I'm actually the
yeah,
same exact way.
I've been totally paranoid about I've been paranoid about that concept of someone actually giving me spice.
Oh, yeah, no, exactly. What
Because I'm
about
like, "Oh."
CBN? I don't think you talked about it, but is that
So
I mean
CBN is an oxidized product, and so oxidation occurs when the plant ages, and so the plant doesn't make CBN, but if you take THC and oxidize it, you can get CBN. It has been attributed to people having better sleep. However, there's a really good researcher out of Israel, Dedier Meiri, who has gone through this, and it's turned out there's two other oxidized cannabinoids that are underappreciated that CBD is just a canary in a coal mine for. So if you
Oh,
have CBN, it's usually a sign that these other cannabinoids exist.
right.
He's called them go to sleep one and go to sleep two, GST one and two, that he presents on, and he's isolated those, and he's been doing studies of those on mice and on humans right now. So it's a good marker. Like if you see CBN, it's likely that those other compounds are there because they are made at trace amounts, and once they get oxidized, they're more potent than CBN. But I know a lot of people who get CBN gummies and they swear by them, that they help them sleep and it works. So yeah, but it can have some psychoactive activities to them if you take it during the day. I've heard people say it's not it's not like CBD where it's very difficult for you to sense any psychoactivity. I think I think the idea of psychoactivity needs to be kind of revisited. There are CBN is an anxiolytic,
Mhm.
and I think for people who have anxiety that take CBD as an anxiolytic, that elation of like the relief from anxiety actually for them is euphoric, slightly euphoric, not like THC, but so the language around psychoactivity we're very careful with in the cannabis space because there are things that are psychoactive, but not that have a neurological impact, but don't necessarily give you the high.
Right.
And even the high in THC is something that you can regulate with pregnenolone. Like if you give people pregnenolone, the high goes away. And the way they monitor this in mice is with these maze studies. And so there's two pathways off of the CB1 receptor when you hit it. There's like there's a COX2 pathway, and then there's this pregnenolone pathway. The COX2 pathway is more related to the short-term memory loss, and you can blunt that with ibuprofen or other COX2 inhibitors like CBD.
Mm.
So people who lose their keys, if you're taking THC and you think you're getting short-term memory loss, ibuprofen or CBD should blunt that. Blunt is the wrong term, but And the people who don't like the psychoactivity, some people will use certain terpenes that could potentially limit this. And beta caryophyllene is one of them that's in black pepper. But pregnenolone has been shown in some science papers that like it shuts down that pathway. So
Hmm.
they're not they're they're different pathways that get triggered inside the cell that result of some of these different symptoms that people report with THC.
Well, that's interesting. So if somebody's having a bad time, they could use pregnenolone to help alleviate that?
Yeah, I don't know how to administer it. And the study that they did in mice, I can't remember if it was IV, and I don't know how well that goes to the GI. But
Okay.
so that's a good question. Jack might know, a physician might know if they've given pregnenolone the different routes of administration for it. But I do know a lot of people will take beta caryophyllene capsules to try to as a reversal of
Huh.
THC.
That's interesting.
So,
It's reminding me of like the trip, like you can use Xanax as a trip killer for psychedelics.
no, yeah, okay,
It's like a
yeah, that
similar concept, but yeah. Yeah, that's interesting. So for the cancer patients, are there specific products or modalities or like routes of administration that you've seen to be more optimal than others, or is that still kind of fleshing out?
so the oils seem to be the best route of administration for that. Now, that only works to some extent if they're not having sometimes the nausea and the vomiting we catch you have to deal with vaporization because they can't keep the drugs down and then you don't know how much they're taking. I've gone through that with my father who had prostate cancer, and we had to use when his the problem with his traditional Western medicine course here was that he was having nausea and vomiting, and so they gave him Casodex, which has a number one side effect of nausea and vomiting, right?
Great.
And so, you're stuck there as a patient in a complete quagmire of, am I getting better or am I getting worse? Because the drugs you're giving me mimic the symptoms of the disease. How do I know? Psychologically, it just wears on you. All right. So we went in and addressed that first. It's like, we're going to get rid of your nausea and vomiting with cannabinoids, and then we're going to see whether, this Casodex actually does anything. And that worked like a charm. All of his energy was back. He was eating like, full salmon dishes before when he couldn't keep applesauce down.
Wow.
It completely reversed the nausea and the cachexia. You need both. You need THC and CBD. There's differences between the feeling of nausea and the actual vomiting. I know they're linked in most people's minds, but they're different. They're different pathways, and each of those compounds have target them differently. So we were giving him very low amounts of THC because he was kind of a novice user, and it was interesting in his case when his PSA was up at like 170. It was already meted out to the bones, so he had tumors everywhere, and we were putting him on about 30 milligrams of CBD, 30 milligrams of CBG, and 30 milligrams of CBDA. So three of those oils. We get those oils from like those epilepsy companies that make a lot of these nice oils. I think Charlotte's Web is one, and Miriam's Hope is another, but they've been in the business for a very long time making making oils for kids. So they've been QC'd by a lot of different labs. Um, and then we were doing that 3 times a day. So that's a high dose. We didn't start in there. You kind of inch your way up over a week, getting up to those levels. So close to 100 milligrams of different cannabinoids of CBD, CBDA, and CBG 3 times a day. All of his nausea went away. He was able to eat. His PSA dropped to 6.
Wow.
And when his PSA dropped to 6, his he started getting high, and that was something we didn't really expect. And I think what happens is if you read the literature, prostate cancer cells express a lot of cannabinoid receptors. I feel like they're called PC. I think they're PC1 cells or PC3 cells. They express high numbers of cannabinoid receptors. So if he had a lot of those over his body, they're soaking up probably all these cannabinoids. The tumors go away. Now there's no mop,
Yeah.
and THC started getting to his brain. We were only giving him like 5 milligrams of THC at night just so help him sleep. But once his PSA started to drop, he was getting stoned by it and was like, all right, I'm going to back off on all of this stuff. And we should have just backed him off the THC and left the maintenance dose and the other things. And then I think when it lasted about 4 years where he was sort of, I wouldn't say totally disease free, but comfortable and living and able to move around, drive around, and live his life. They had to completely remove all the cannabinoids when they wanted to replace his pacemaker because his pacemaker was not MRI compatible.
Mm.
That was a surgery, and the anesthesiologist didn't know how the stuff was going to react. So he's like, just dry him out for a couple days beforehand. We'll go through the surgery, and then you can put him back on afterwards. So he came out of that surgery with vomiting, screaming, coming back. And the hospital couldn't contain it, and we finally told the nurses we have something we can use to get rid of this. And first asked them if they had THC, like dronabinol, the prescribed stuff, and they didn't have it. We gave it to him in 30 minutes. He was up, walking, ready to leave the hospital, and all the nurses came running like, "What did you give him? We have nothing that could stop that, and we want to know what it is." They started taking pictures of all the bottles, everything.
Wow.
And the doctors didn't ask a thing, but the nurses were like, "Yeah, that's important. We got to know what that is, so we can help others." Um, so that's when the therapy started to fall apart for him. Is we went through that sort of bleed out of him using those things, and we were doing sequencing of his bloodstream periodically, and monitoring the mutation that he had. He had a particular mutation in his prostate tumor that was a K601E variant
Hmm.
in shit. What gene was that in? It's a BRAF. And so the that we could see going down in the blood over time as well. And then he got a P53 mutation after that time, that period where we pulled him off all these things. They
Wow.
were also playing around with other chemos at that time as well, which may have played a role. But once the P53 thing came into view, Dana Farber kind of freaked out and was like, "All right, we have to accelerate. We got to drop everything on this thing." And and then thrombocytopenia showed up, and it got out of hand pretty fast. So we don't have a good control here. I don't know that he would have been able to survive all this on only cannabinoids, because it was cannabinoids supplemented with what the sort of standard regimen hormone therapy was, which was very bizarre to me, because that hormone therapy is really meant for people that have androgen receptor. Like 90% of prostate cancer is an androgen receptor mutation, right? And he didn't have one. He
Mhm.
had what looked like a melanoma mutation. But they're like, we have to start with the. It's kind of like, they won't let you get an MRI unless you get an
Yes,
X-ray.
it's the
You
insurance
know,
middlemen
yeah,
bullshit.
exactly. And you're like, wait a minute, we just did all the sequencing that shows that's not where we're starting from. We should start over here on AKT inhibitors, right? And they're like, no, we have to go through doing Casodex and rule all these things out, and then we can graduate,
Mm.
torture the patient with nausea and vomiting along the way, and then maybe we'll consider the sequencing that you guys did.
Wow.
Um, so it's a little bit frustrating. It definitely opened my eyes to like the world of cancer, and cancer therapeutics is dreadful, and that the rules go out the window with terminal care. I think there's even odd conflicts that can occur with physicians and what they prescribe, and there's these regimens on certain cancers where they have to roll you through a torturous protocol and rule things out before you can get to what you really need. But I've met very few people who have not been at least gotten prophylactic, or I should say, not prophylactic care, but the dealing with the symptoms of cancer. Cannabinoids work. Like I've heard this from everybody, that when they have pain, nausea, and they can't sleep and need to eat, the cannabinoids alleviate a lot of that pain. And you don't have to get on morphine drips that get you constipated and run you down a wreck. He never, even having bone meds, he never got a morphine drip. So it was we got the pain under control with just the cannabinoids. But whether they actually shrink the tumor is where there's evidence for this in some cases, but I don't think we fully understand like which tumors need which cannabinoids.
Right.
Like the glioblastomas. We get some people report very low dosages that work. Other people are using really high dosages that work. That's still an unknown domain. I think what's exciting about them is that there isn't this risk of overdose. So you can experiment with a broad range of dosages. And the running advice in the field is start low and go slow and ramp them up until you're comfortable. And when your body's comfortable, you're at the right dose. Do not try to exceed that to try to fight cancer that you can't feel. Like it's really, your body will tell you when you have the right dosage of these, and leave it at that dose for as long as you feel necessary. And you start to see other signatures of the disease disappear. If there's other biomarkers you're tracking, you should see those, watch those in concert with the dose that you're at.
Speaking of cancer treatments that are kind of taboo among centralized medicine, I'm thinking of Dr. Mackis's work with ivermectin, fentanyl,
I'm very
all. What'd you say?
Yeah, I'm very interested in all that work.
Yeah.
And I don't. I'm reading on it. I can't. I have no experience with it myself. I'm not a treating physician, but I've been reading about all the case studies. And I did. Yeah, I dug into some of the literature there. I was intrigued in that the ivermectin looks like it actually blocks S40 from getting to the nucleus. So
Mm,
okay, that could be interesting. And then fentanyl, and I think they're replacing it with another fen version of the drug that's a little bit better. But that thing stops, I think, microtubule formation, so that fast dividing cells have a hard time dividing.
right.
Uh, and I believe there was even some BRCA1 interactions in p53 interactions from ivermectin or fentanyl. I can't remember which one, but it made sense as something that would help if this vaccine-based cancer thing is a result of any of this DNA contamination or is a result of the spike protein inhibiting p53. It made sense that those pathways should be considered. And the drugs, it doesn't seem like those drugs are like cannabinoids. You have a large dosing window. It's not like you're threading a needle between toxicity and therapy with those drugs, like you are with cisplatin and a lot of the other cancer drugs. So it seems like it's completely worth investigating and worth a try. Um, I've seen other people say, it doesn't work, and of course, if you put them down that path of hope and they don't take the other drugs, you're actually killing them. You get into all those bizarre zero sum fallacy arguments. I think a lot of people who
Yes.
don't want to consider it, but I hope to see more physicians, experiment with those in cancer because right now Macus is under a lot of fire, and they're going after his bank accounts and everything
Oh,
else,
yeah.
and just it'd be good to see some more replication of others, others having success and more. He's at least very good at publishing a lot, putting forward a lot of the papers that are coming out. So it's not just him. He's just been an amplifier of it, I say.
Yep.
Um, but I'd like to get more input on that. In fact, those are areas that I wish we had those tools back when my father was going through this because those may have helped him in other ways, hitting other pathways that cannabinoids can't touch.
Totally, yeah, and we know also there's some data showing that ivermectin can help, activate autophagy, which in combination with other things can help on the
Yeah,
cancer front to help slow
yeah.
down growth and glucose metabolism and all those things. But speaking of, this is maybe a good time to start talking about, like, the plasmids and the estrogen promoter and such, so do you wanna, like, talk a little bit about how you even ended up stumbling upon, this issue, like, that promoter was present in the Pfizer jabs, and I heard somebody say that there could be some in the Moderna jabs. Do you wanna update us on that as well?
Oh yeah, so I haven't found any of the Moderna jabs. That was coming from I
Okay.
think Chakrabarty's work, and I think that's an artifact from. It gets into some of the weeds of how DNA sequencers behave. When we're sequencing a lot of samples on DNA sequencers, we tend to put DNA barcodes on them to sequence them because the sequencers put out like terabases of information in one like $20,000 run. So what happens is you get 100 samples you want to put onto one run, you got to barcode them somehow. And the way we barcode in genomics is we take a piece, like an 8 base pair piece of sequence, and glue it to that library that we're going to sequence. And when we sequence that as the first 8 bases, we can say, okay, that's an index, that's sample one, that's a barcode that says it's sample two, sample three, so on. So the problem with some of these sequencers is that they can have a barcode misreading rate of around 1%. So 1% of the reads that they're reading as barcode one get miscalled as barcode two, and suddenly you have what looks like chocolate and peanut butter. And I think that's what caused the issue with what Chakrabarty was looking at. It wasn't his data. He was reading data from another laboratory and going through it, and the methods weren't really clear on how they handled the barcoding, so he just wasn't attuned to that. But um, so in that case, yeah, you technically can find some in some of those Moderna samples from the Ryan et al. Paper. But when I've really tried to scrutinize those, I think that's actually a sequencing artifact from barcoding. So I've not yet seen credible evidence, and we've done qPCR on hundreds of vials now that there is SRP in the Modernas. Doesn't mean they won't use it in the future, but it's not there now. So all right, how did I find this stuff? I was doing RNA sequencing of cannabis plants as they're being infected by hop latent viroid. And that's an RNA viroid. And in doing this, you have to extract RNA and sequence it. And one day, that process was coming back. So usually when you do that, you get sequence coverage across the genome over all the genes, and you get all these gaps of sequence where there are no genes. So it's a great way of like painting on the genome where all the genes are. Is you read the RNA and then map it to the genome, and you can see where all the genes are. Well, one day our informatics team came back saying, "Look, this last run you did, the reads aren't concentrated on the genes. It just looks like whole genome shotgun. Something screwed up in your pipeline. You need a control, spike in a control RNA, and just prove to us that the beads you have that are capturing the RNA are doing their job. And if that RNA comes through, then we'll be able to pinpoint where in the pipeline something's broken." So I was like, all right, that's a great idea. I don't have an RNA handy except for these RNAs someone sent me that look, they're vaccines. They have a poly A tail. I'll use those. Will that work? And they're like, yeah, it's perfect. It's, pharmaceutical grade. Spike it in. So I do that, and it comes back, and I'm like, so what's broken? And they're like, well, your RNA came through, but so did all this other plasmid DNA. We don't know what this is. Did you put that in there? And I'm like, no, that's got a spike sequence in the plasmid, and it's got SV4. Oh, wait a minute, that's their
Oh.
expression right there. And I was like, oh shit, this is the problem I didn't want to inherit.
Oh.
So immediately I was like, oh, this is such a headache because I know I have to I can't hide it. I got to put it out, but if I put it out and I put it out sloppily, I'm going to be drawn and quartered. So I asked the team, okay, why don't we just go and measure this every way we can in the lab? What do we have? We can measure with the Oxford Nanopore. We did that, which we sequenced it with another sequencer. We did it with Illumina. We did RNA sequencing. We did RNase. We erased all the RNA and sequenced it to prove it wasn't like some artifact of the RNA. We ran it through an Agilent bioanalyzer. We ran it through a nanodrop. We ran it through a qubit. We ran it through qPCR every which way to Sunday with DNases and RNases. And lo and behold, it's there, and it's a way higher copy number than anyone would imagine. So reference points here. A good reference point. You probably got a PCR test, or someone in your audience may have gotten a PCR test for COVID, and it probably stopped calling you positive at around 35 or 40 CTs. And this thing comes in at around 15 CTs. So for those not familiar with that offset, that's a 20 CT offset. That's a millionfold more DNA that is, as a contaminant, is being injected into you with LNPs than what they're scraping out of the outside of your body in your nose with for viruses. So for them to have a hyper concern over a replicatable virus that is a millionfold lower concentrations than a replicatable plasmid that's a million times more concentrated in LNPs than being injected is complete idiocy. Like, they cannot defend this. And the way that they try to defend it is they say the plasmid's not replication competent because you chewed it up. I'd love them to prove that, because when we've FOIA'd Pfizer about this and Health Canada about this, they have no assay for the length of the molecules that are in there, so they don't know how well it's chewed up.
Mm-hmm.
When we've done Oxford Nanopore sequencing on it, we get like 3,000 base pair fragments that come out, reads that come out of, like, when we just do like 800 reads. So if I do 8,000 or 800,000 reads, I'll find one that's 7 kb that's replication competent. I'm sure of it. How much these things vary lot to lot, there's got to be one that's replication competent that's in some of these files. I don't know that's in all of them. They are trying to erase it, but the way that they're erasing this DNA is asinine. They're not using the right enzymes to get rid of this DNA. It's a solvable problem, and just the mere fact that there's more of a problem in Pfizer than there is in Moderna proves to you that someone in the group's being lazy.
Yeah.
And it's not a small difference. Like, I think Moderna is probably tenfold lower than Pfizer. So,
And that's even given the fact that the Moderna dose is like double the dose of,
yeah,
like,
exactly.
yeah.
Yeah, they're like triple the dose. So,
Yeah.
Moderna actually has patents out there that describe this problem. They actually invented a new purification method because they're so concerned about it. Their patents are like, this stuff is oncogenic. It can do insertional mutagenesis, so we have to do a really good job getting rid of it. And so they come up with some new method for getting rid of it. And clearly they're doing a better job than Pfizer. But you can put the data in front of these regulators being like, look, you've got one manufacturer in here that's like tenfold lower than the other. And you're trying to claim that it's acceptable because one group has tenfold more than the other. Like, why is it acceptable? If it can be done one way, why can't it be done for everything? They're not very well versed in this stuff. I mean, so the reason this is happening is when you have RNA inside of a vial that has the DNA it was made from, that RNA is hybridized to that DNA. That's the synthesis process, right? An RNA polymerase goes through and copies the DNA and makes one strand of it RNA. You have RNA hybridized to DNA. That is thermodynamically more stable than DNA hybridized to DNA. Just for folks who understand the nucleic acid chemistry here, RNA sticks to DNA stronger than DNA does. So these RNA DNA hybrids, the enzymes don't know how to erase. At least some enzymes. DNase one does not erase RNA DNA hybrids. So the region of the spike sequence, that's the region that's not getting destroyed, which is why we think it might be related to the longevity of spike being expressed.
Right.
Because that's 100-fold higher concentration than the other part of the plasmid that the DNA is double stranded. There isn't any RNA, and it chews that up really readily. But they let pharma pick a PCR assay that targeted the part of the plasmid the enzyme can kill. Very sneaky. And the part that actually is 100-fold higher in concentration, they had an assay to measure that to prove that the spike sequence was inserted in the plasmid, but they refused to use that to quantitate the DNA because they know the answer it's going to give them. It's that they're
Wow.
100. So this stuff is circulating, and you can use a different nuclease to get rid of it that does process RNA DNA hybrids, but they're not using it. So there's a known way to solve the problem. They're 100-fold over, and there's other manufacturers out there that have the problem better addressed, but not perfectly. And all of this DNA contamination is happening in an environment where now we have lipid nanoparticles present that bring it right to cells. Like
Right.
all of the boundaries and regulations they have, and that we can accept 10 nanograms of DNA if you inject it. That's all because it's injecting it like naked into your arm, which has DNases to chew it all up. But once you put an LNP in there, that's the reason the mRNA platform works, or supposedly works, right? That's what delivers it to the cell, and it's dragging all the DNA with it. So now, pharma likes to run around and be like, "You've never proven that this inserts into anyone's genome, so we're going to now shove it into babies, right? The burden's on you to prove that our stuff
Oh,
integrates." And we're like, "No, no, no. It's integration is like, yeah, that's a horrible thing if it happens, but the moment you put DNA into the cytosol of a cell, you trigger an interleukin response. You trigger a cGAS sting pathway. You trigger cancer. Like you can't repeatedly hit cells with cytosolic DNA and not make the cell think it's infected with a virus. All right. So what it's going to do is it's either going to trigger apoptosis and destruction by the immune system, or it's going to go into oncogenesis and it's start growing rapidly. And there's papers from Quan and others that describe this. So the insult isn't whether it messes with your genome. Of course, that would be horrifying if we find it, and there's people working on that. It's a harder problem to solve. The insult is the moment you flooded cells with cytosolic DNA, you've thrown off the immune system, and that can be a contributor to cancer. So they don't want to talk about any of this, and it's they've been running around saying you eat DNA all the time. Don't worry about it. DNA's in tomatoes.
oh,
You're eating DNA right now.
that's so
And
disingenuous.
it's like it's an insult to the intelligence of the American people that like, oh yeah, eating drugs is the same as injecting them. I mean, is that why we have people all over with heroin needles in their arms all over the streets? I mean, it's asinine that they're floating these gaslights that I suspect, I guess some people are still buying, or I don't even know how many people still believe in these things. I just know people are forced to take them. That's the stage that we're at right now. Is that
Mhm.
the kids are getting it's getting forced onto the kids. I don't know a lot of adults that are willingly taking them.
Oh gosh. Yeah, and I remember actually funny enough when the jabs rolled out, I was at Princeton still and everybody was first of all just so excited to line up and so excited to get it first and Pfizer was seen as like the good one. Like everybody wanted Pfizer at school and like, "Oh, not Moderna, Pfizer, please."
I don't know how that happens. I guess they have a bigger marketing budget than Moderna. Maybe it's odd. A good person to chat on that topic is. Have you spoken to Jessica Rose yet?
No, but I've heard her on a few podcasts. She's great.
Yeah, she's got a good sense of the adverse events. And so I don't know that there's substantially more or less from Moderna or Pfizer. It's hard to untangle because Moderna has more RNA, which is a problem in of itself, and different LNPs than Pfizer. So you're getting like triple the dose there.
Yeah.
So the fact that they may have less DNA, but you end up tripling the dose is getting closer to being similar. But still, I think in total, dose by dose, you'll get less DNA contamination with Moderna. They have more double-stranded RNA though, from my assessment of the two different vaccines, and that has its own like stimulatory pathway and the toll receptors that could wreak havoc. Um, and then the Pfizer stuff though, I've seen from what is Marianne? This Marianne Latteratz did some of this work where she looked at the onset of the tuberculars and was finding that Pfizer had a faster time to death in those cancer patients than Moderna, but
Mm.
equal amounts of cancer between the two of them. So they both can be oncogenic, but Pfizer seemed to have a faster expiration, if you will, the way it was termed in that. So um, they're different. There's a lot of things wrong with these things. I think if you get rid of all the DNA, it's not going to solve the problem. So we're not trying to pin everything on that, but the DNA is just one of these like traceable markers that we have a hard time tracing the LNPs. We have a hard time tracing a lot of the other components in these things, but the DNA is something we can amplify from single cells, and it's a forensic marker for their mistakes. So it's something that can be measured in a decentralized manner. Like PCR is everywhere. You can PCR in your basement, right? So if they can run PCR in parking lots to track COVID, they should be able to run PCR to track this contaminant in all the patients that have biopsies that are have questions about them. There's others out there that are looking at spike as well, which is important because there are cases where we're seeing spike out there like 700 days, like we saw in that Yale study. Bruce Patterson's lab is probably the pioneers of that. They actually had data on that long before Yale, and I think at Incel DX they're building tools to look for viral spike versus vaccine spike because they can differentiate them. But yeah, we've run into some biopsies where we can barely see the DNA, but there's shitloads of spike. And so
Wow.
I don't know what that means. I don't know where that spike's coming from and why it's still there, but it's maybe it's getting traffic from some other cells we haven't sequenced. But we can't just look at the DNA alone. It's only one part of the story.
Yeah, and isn't it the case that Pfizer left the fact that they used an SRB promoter out of the patent that they
They
submitted?
did, yeah.
Yeah.
So there's a submission to the EMA, and there's guidelines at the FDA and the WHO where you have to disclose every open reading frame in the plasmid and every promoter, and they hid SRB from them. And I say hid because I know they did it, and that when you load up their sequence into commercial plasmid annotation software, the first thing it annotates is SRB because it's one of the most common biotech tools for hyper expressing a protein. Right? It's just if any of you are familiar with plasmid work, it's like M13 sites or multiple cloning sites. These are sort of the common units that are in plasmids, antibiotic resistance genes. So these tools will readily annotate that if there's a kanamycin resistance gene here, there's a T7 promoter here, there's a CMV promoter over there, and there's an SB thing over here. So their plasmid map got annotated by something that mysteriously erased the most commonly annotated region of a plasmid. So someone had to manually remove it. There's no other explanation for that. There's no way a software tool would have annotated the kanamycin resistance gene and omitted its promoter. That's not possible from a software standpoint. Someone manually deleted it, handed it into the FDA, so the FDA wouldn't be aware of it. And then, because had they been aware of that was in there, they would have realized that's the mammalian promoter. You've got a vaccine with a plasmid that can now replicate in humans. You don't need that. Moderna doesn't have it.
Huh?
Why do you need it? All you need is something that replicates in E. coli because you're manufacturing this E. coli. So make sure there's nothing in your plasmid that could replicate if it ever got into a human host. Now that part is actually a bit frightening because there's now papers from Beck in Seattle who showed that the vaccine plasmid they were working on, which had an SRB promoter in it, um, it was called pcDNA 3.1. That's one of the most common plasmids used to study spike protein in parts of this virus. These guys were studying nucleocapsid. Turned out the whole lab got infected with the thing, and it got outside the lab. Had that been Spike, they could have been responsible for spikopathy spreading out, from their lab. But that's the artifact of having mammalian promoters in there. It was it was coupled with a bacterial one as well, so it could replicate in both bacterial and so they had an origin of replication and a bacterial origin of replication. That means it's a shuttle vector. You can replicate it in E. coli and in mammalian cells. And they were hypothesizing that this must have been replicating in E. coli in their laboratory, and then someone got it on their hands, and E. coli loves their skin, so it got all over their nasal cavity, and now they're picking it up in COVID tests in all the people in and outside the laboratory.
Wow.
Uh, so they need to redefine gain of function research to like they got to put some control on these shuttle vectors that can bounce between E. coli and humans that are carrying these viruses that these toxic viral peptides. But that's effectively where Pfizer's vaccine came from. Is it looks like a pcDNA 3.1 plasmid that they got from their gene therapy department, and they didn't bother to rip the mammalian origin of replication out or the SNP components out and just ran with it into production. And now it's in the vials, getting into patients, and we have another mess in our hands like we have with the polio virus. Now, it's not the full virus, but it's some of the parts of that virus that allow the DNA to replicate in mammalian cells. It's got an aggressive promoter that anything that DNA lands in front of in the genome, it's going to hyperexpress. It's got a nuclear localization signal in it, and it's also got this hypermutability element in it. So there's papers out there showing that the enhancer is a hypermutability element. It encourages mutagenesis in DNA that's near it.
Wow.
There's all these components to it that are just a train wreck, even without it having the T antigen in it. So that's what they try to deflect on, is saying, look, we don't have the T antigen in there. The tumor antigen is what causes tumors, right? So we don't have the tumor antigen in it. It's like, no, I don't think you understand how viruses behave. Like the tumors can be caused without the tumor antigen if they insert in the wrong place or they just trigger the cGAS sting pathway. So um, it's very, very sloppy molecular biology that they're leaning on to try to gaslight people. Like physicians in the industry, love them, but they don't have this level of detail on like how these plasmids work. I've worked with on the Human Genome Project. We built tools to purify like 20 million plasmids. Like I live, eat, and breathe these damn things. Each one of these components in these plasmids has a distinct function inside of a cell. They're designed and they're engineered into these biotech vectors for very specific purposes, and the pieces they have in there are gene therapy components. There's just no question about that, and they left them in there.
You think it was laziness?
Um, that would be a kind way to describe it. Yeah, is that okay? Maybe it's just lazy incompetence, race to market. But when I saw that they started buying cancer companies, like a year into the pandemic, Cgen, $42 billion in cash dropped. Another one was Trillium. Trillium was targeting blood cancers in particular, which is what's happening with this. I start to question whether that was it really laziness or was it like we have no liability here. Let's create a market after a market, which is how will anyone ever trace this back to us? They'll be able to say, well, you found some STD here, but it's been around since polio. Sorry about that one too. But how do you know it's ours? I mean, people are running around PCRing for STD in these biopsies, and we're finding it on occasion. But that does raise the question: were they vaccinated with the poliovirus? How do you know it's theirs, right? And so there's a little bit of an out for them in that regard. Now we do have ways we can figure out it's theirs. If we see spike, it's them, right? The spike is not from polio. And the spike sequence is so uniquely theirs. It's a smoking gun fingerprint that it came from Pfizer and Moderna, and we can differentiate those. But when we get things back that just have STD and those are missing, it's a little bit more of a okay. We can't. We've narrowed it down to one pharmaceutical asshole, but not which one.
Right. So are the tumors with the STD only from people who are old enough to have received the polio vaccine with the contamination?
Well, that's a good question that I'm not the best person to ask, but when I have read into STD, it appears it was spreading beyond the people who were vaccinated. So when they ran
Whoa.
the polio vaccine and you got the full STD virus, it would then express and shed. And so even the people who were not vaccinated were getting it. And that is one of the reasons why I think that debate still rages on today. If you put
Mm.
STD inside of mammalian hosts, they get tumors. When you try to find a split in a demographic where vax versus unvaxxed, you can't find it because it's shedding. And so you don't get a clean separation of who got cancer and who doesn't get cancer because some of the vaccinated gave it to the unvaccinated.
Right, that makes sense. What do you think about shedding with regards to the current mRNA vaccines?
It's so Pierre Corey has a good thread on that. I've been reading his work on that and Western was the Midwestern doctor, I think. They think it's happening. I want to find some molecular tools to prove it. They've
Yeah.
gotten good clinical like arguments for why it's happening, like the menstruation occurring with women and your
Yep.
proximity to. I mean, they've got a lot of good arguments why it's happening. What I've always been trying to find is what is the molecular mechanism? Because if you had shedding of just spike through exosomes, let's say, you would think it'd be such a low dose that it wouldn't matter, and then maybe you'd only see it in hypersensitized people because there are people out there that are very sensitive to spike, and they get near it and they flare up with rashes and what have you, and so we have to be cognizant of the fact that maybe a low dose can trigger those people, but I always assumed it'd have to be something replication competent. Like if the plasmids were moving in exosomes into patients, that would explain it. That okay, you only need one to get in and then it takes off, right? Um, but I don't have proof of that. I've only seen this spreading event happen in Seattle to be like, well, it happened with the backbone that Pfizer's using. I mean, the vector they have in that Seattle study is pretty close to what Pfizer has. It's got many of the same components. It's just nucleocapsids in there instead.
Right.
And that thing replicated throughout a population and spread from the lab and out. So if there's any full length plasmid in there and it may not be in every vial, maybe it's in some minority of the vials that are really bad, they didn't digest well, and then that thing gets out and spreads amongst the community, then that could explain it. Because the exosome data is you breathe out exosomes, so your body, when this thing gets into you, the cells want to get rid of it, so they package it up in exosomes and just it's like the garbage collector, get it out of here. They send it out, and then your body starts exhaling those, and it comes to your skin too, out of your sebaceous glands in your skin. And that's one way we can get contact, perhaps exposure for this. So, the jury is still out. I haven't seen like a hard paper proving like, all right, this RNA went from this person to that person, or this DNA went from here to here. But the fact that it's persisting for so long in some patients, and then we've got all the work that Corey and this Midwestern physician put together showing that these are all the signs that it's not hormonal and that there's actually something related to the vaccine that's moving around, leads me to believe it's happening to some extent. It's only going to get worse if they do these self-amplifying RNAs.
Oh, yeah. I want to talk about that. Is there any precedent for having plasmids released from the body via exosomes, like in other contexts, or not that you know of?
That's a good question. I would assume that they would get moved because they're small enough to move. There is evidence of DNA traveling in exosomes, without a doubt. In fact, that this is the basis for why they think it's the mRNA is ending up in breast milk.
Mm.
So there's a couple studies from Hannah et al. That look at breast milk, and um, they that's a bit of a paper to read with caution. Like just put your DEI filters on because they talk about pregnant people in that thing.
Ah,
Uh, and so but they also try to claim that this is a minimal amount getting the breast milk. Don't worry about it. But when you look at the limit of detection, their qPCR assay, it's like at 40,000 copies or something. It's 400,000 copies. It's horrendous. It's the least sensitive PCR assay I've ever seen. It should be down at 10 copies. And so they have this really like blind PCR reaction going in saying, oh, look, we only find so many nanograms and below that we can't detect it. And it's like, well, that's because your lot is orders of magnitude too high. You're an idiot. And if you account for the fact that you could pick up 10 molecules with a normal PCR reaction, you're feeding these kids pretty much a human. If you weight adjust their weight and the amount of milk they're getting, you could argue they're going to have a full vaccine dose orally taken within a couple days of feeding. And those now people say, so what? It's going in your gut, you're eating it, but it's coming in breast milk, which are extracellular vesicles. Those are exosomes. Those are transfection vesicles. So everything your breasts are feeding to the child is getting transfected into their mouth and their throats, and that's just how the children build immune systems through their GI. So these extracellular vesicles survive the stomach, so they get to your intestines, which is part of the reason, part of the mechanism of how your mother passes immunity to the child.
mhm.
Right. So for them to be like your GI is going to destroy this stuff, and they're picking it up in extracellular vesicles, these EVs, means that they're lying. Like they're totally lying about that story because everyone in that field knows that that is a communication channel to move immunity to your child is through breast milk, and whatever if you're packaging the mRNA molecules into those things and the DNA, that's getting into the kit. And there was a study, I think it was who was the first author of this thing? It came out very recently where they showed it getting through placenta and into the newborn in mice
Wow.
and expressing spike protein, and they can detect the mRNA and the spike protein in the newborn like 7 or 10 days later. So um, yeah, when it comes to shedding, it's clearly shedding if you consider placental transfer. That's clearly a shedding event. It's clearly shedding in breast milk. Now, the question is, what is the dose people are getting through skin and through exhaled exosomes? That's the question mark that I think has not been quantitated very well.
Right, and especially also as it relates to, like, adverse events, whether it's from shedding or from directly receiving the jab, I think, we have no idea the implications 1, 5, 10 years down the line, right? Because 'Cause they're only looking at, a certain period of
Yeah.
time, and there could be many different effects that they have plausible deniability for, of course, but, they could show up down the road, like, maybe we're shaving years off people's lives, who knows?
I think we are. I mean, the if you look at the Japanese study from Gaibo et al., their life expectancy is decayed for the first time since like Hiroshima. I mean, it's that the graph that they show is absolutely horrific. They have had more excess mortality than the tsunami in Hiroshima combined
Wow.
since these went in place. Now it's not all cancer, but it's like thrombotic events and acute renal failure, a bunch of things. I think the cancer is only like maybe 5 to 7% of the excess mortality, but it's still risen, and they're all in cancers they didn't have before. Like,
Right.
what's fascinating about that paper is they show the cancers pre-vaccination were of these flavors, and then afterwards they shift to being like ovarian, lymph. Um, uh, I think there was prostate in there as well. So the profile of the cancer types they had before and after vaccination program shift, and the age demographic goes down. And it's not it's the reason they don't that paper's pretty clear that this is not the virus is because they got so many goddamn vaccines. They got like 123 million vaccines. It was something like they got for every person that got infected with the virus, they took 9 vaccines.
Wow.
Yeah, I mean, think of the economics of that. You're taking 9 of these things to prevent a coronavirus infection, and so yeah, the coronavirus infections are hard for them to gauge on like how actually deadly were they because they had such a high toxic burden of vaccines.
There's no control group. Yeah.
No. Other than the tight window, there's one window where, the pandemic is occurring prior to the vaccination getting in there. It's a very tiny window, but almost all of the adverse events are temporally related to after the vaccination window.
Mhm.
So if you take that paper and you plug it into a ChatGPT and feed it like I've done this. I have a Substack on this, and you feed it with a bunch of papers. If you feed the paper initially in a ChatGPT, be like, "Well, this does not really fit Bradford Hill criteria for causation because X, Y, and Z. It's correlated, right?" And then you give it like 5 more papers. It will change from being safe and effective language to "Holy shit, you need to pull these off the market."
Oh my gosh, that's amazing.
Yeah, you can watch it. You can watch ChatGPT go through the logic of being, of being red pilled on this thing, being like, okay, in light of those papers, this is the problem, and they need to emergency recall right now.
Wow. Well, actually, speaking of Japan, so when I was at Rescue the Republic, I think it was in October of last year, that was the same day that there were these, like, riots or protests against the self-replicating mRNA jabs. Do you wanna briefly talk about the difference between those versus the current.
Yeah. So those are a nightmare. I mean, they're it's a really bad idea. They're trying to make the claim that we need a smaller dose of the RNA to get in and make a hell of a lot more protein, so we can make these safer by lowering the dose, kind of thing. And they've got no evidence they need the high dose that they have now, and that the dose is the problem. It's arguably the protein that they're expressing and the transfection that they're doing. It's the problem. But they use some encephalitis viruses, components of them, an RDRP gene to do this. It's an RNA polymerase, an RNA dependent RNA polymerase that basically will help replicate the virus once the plasmid once it gets in. The problem with this is that that sequence signal that it uses to make that polymerase is in other viruses out there, other
Mhm.
EVs that exist. There's some in South America, there's some in North America, and that means if you get a co-infection of someone who's vaccinated that also has those viruses, you don't know what the hell's going to happen. It's very likely that you're going to that vaccine is going to trigger those other viral infections to go crazy because you basically have a signal for the virus to replicate that vaccine component whenever it's present with one of those other viruses. And there's a lot of these other viruses out there, these other EEVs. So I think it's a reckless. It's as reckless as like Bill Gates trying to use mosquitoes to spread drugs. Or it's just they're way over their skis. It's hubristic that they think that this is a good idea. It's about patents. It's about them. The academics are all cheerleading this because they're probably on the payroll of the patent state that's involved in this, and it's out of control. That's a horrible idea, particularly if they're doing this for respiratory viruses, which are treatable.
Yeah. It's also making me think of, I don't remember his name, but a couple months ago he was coming out saying, in the future, maybe the near future, we're going to have these targeted vaccines against certain cancers like mRNA platform or whatever. Do you remember who it was? It was
Oh
ridiculous.
God, that was Larry Ellison, I think,
That's
right?
right. Yes.
Yeah, 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 database guy. Yeah, no, I mean, he's clearly not aware of the fact that these things are causing cancer. So coming, it's like, yeah, we got cisplatin, we're going to use cisplatin to prevent cancer. These are mutagens, if you will. I mean, they're not direct mutagens. They haven't done a lot of work to study the genotoxicity of these things, but there's the investment at the NIH would be far better focused on looking at all of these other off-label drugs that have wide therapeutic indices, like ivermectin and fentanyl and cannabinoids, and even psilocybin. I've seen work on that, like solving depression, terminal cancer depression sets patients down a horrible outcome path. Right, once they're depressed, they're out. So there's been great work at Johns Hopkins looking at psilocybin for rescuing people from terminal cancer depression, and they often have better outcomes. We've seen that with a couple case studies down at CanMed where people used high doses of psilocybin and cannabis oils and eliminated their triple negative breast cancer in 6 months. Like incredible stories like that need to be investigated. But that's a money machine thing. And granted, Larry Ellison was supposed to be the money machine for that, or maybe SoftBank was the money machine for that project. But I don't have high hopes that it's going to go anywhere. These are billionaires that don't know if the first thing about what they're trying to get done and are trying to throw money at the project to get in front of the president and hopefully get some type of government program that puts some wind behind their sails, I think. But yeah, there are some. I'm not going to throw the entire mRNA thing under the bus. Like there are some diseases where it might make sense if you have cancer and it's terminal and this is your only shot. Then okay, that makes sense. Respiratory viruses, the risk ratio is just not there. But there is a case in so Hatter is one disease where they use short RNAs, which are you can synthesize those cleanly without any DNA. There's like microRNAs, right? And when they infuse them over 6 hours with an immunosuppressant. Right. So totally different route of administration, different synthesis process, and the terminal disease. Okay. That different story, right? There's other cases where they've used lentivirus plasmids to treat. They take your bone marrow cells out, and Duncan et al. Has a paper on this in New England Journal of Medicine. You take your bone marrow cells out, you treat them with lentivirus plasmids. They do integrate. They intend to integrate these things. They integrate it like 1,000 times per genome, and when that happens, 10% of the patients get cancer, but they're willing to take that risk because they have a lifespan of 20 years if they don't do something.
Right.
All right. So these are this is where this gene therapy stuff was like designed and marketed for. It was not meant for like, let's take this to as a replacement for ibuprofen or whatever, zinc or whatever you might use for a cold. They're trying to use this because the reason they want to go for that market because it can hit everybody,
Yeah.
right?
Exactly.
When you get into these rare diseases, it's like, well, we don't have enough patients, so now we have to charge $1 million of therapy. So, we got to find a way to get this into cold patients.
Yeah, exactly. And I'm also thinking I have some friends and loved ones who work in pharma, and one of the companies, I won't say who, is working on a pan-coronavirus vaccine, which is insane because my opinion on viruses is like, especially coronaviruses, they're a very staple part of like the microbiome of nature, like of natural environments. They're extremely common, and most of the time they're not causing issues. Sometimes they do, but
Your
usually they're
genome is
pretty.
8% viral.
Exactly, and so I personally see viruses as, like, primary drivers of evolution and kind of like little thumb drives that can come in and give you some upgrades about, like, what's going on in the environment, let's say. To think that we like, in our hubris, that we should somehow make our immune system, think of these things as pathogens when they're actually, serving probably a really important function, and the fact that they don't really cause severe disease, it's just like, what are we actually doing here? It's like you said, we're
Yeah.
just creating a market for everybody.
It's a very good point that the assumed pathogenicity of viruses is it's funny that they how they've done this. There's a couple may help people kind of break out of the spell, right? There's a couple lies that they feed into this. One is this: all viruses are bad, right? But we're going to use a viral vector to cure you.
Yeah.
The other one is well, you shouldn't give out antibiotics to everybody because you'll breed antibiotic resistance in bacteria. But when it comes to viruses, we're going to vaccinate everybody with a narrow vaccine, even though viruses mutate faster than bacteria, right?
Makes no sense.
Hello, this is bio 101. Like they're violating all of these rules to try to fit their pharmaceutical industry into this narrative. But I agree with you in that, so here's an interesting evolutionary hole that was filled just this last year by Andy Fire's group. They found these things called obelisks, right? So, for those not familiar with the evolution of these molecules, viroids are circular pieces of RNA that are short, usually like 250 letters, maybe 300 letters long, and they are they don't code for any DNA or any proteins. I'm sorry, they're just RNA, and they being an RNA molecule, that means that they're a ribozyme. They can catalytically fold and perform reactions inside of cells that for some reason the cells propagate them. And in the case in the cannabis, sometimes the viroids are really pathogenic. I mean, the ones that we track are, because there's an economic motive to track the ones that are pathogenic, but there's probably 100 we don't know about that are non-pathogenic and may even be beneficial. We don't track those because no one cares yet until we can figure out a reason for them to improve the yield in the plant. So what Andy Fire's group found was that this missing link between viroids and this thing is driving me nuts. Let me turn my monitor off because I think it's flashing on your screen.
Oh, no worries.
Um, the evolutionary link between viroids and viruses was like, okay, well, where is the circular RNA molecule that codes for at least a protein? And because we're going from this leap from a viroid which has no protein code, doesn't code for any proteins, but is a catalytic ribozyme. Then the next leap is a capsid enclosed viral genome. There's got to be something that is codes for some protein but doesn't have a capsid. Where is it? Right. And so they bioinformatically went looking through all the RNA data they could find in NCBI and found these rod-shaped RNAs that they called obelisks that do code for one or two genes. They're they're 1,000 letters long. They're hairpinned, and they code for one or two genes that I don't know. I forgot what they code for, but they're important in transmitting replicating this actual viroid. They were only found in the oral microbiomes of healthy patients.
Whoa.
Right? So you're like, okay, the evolutionary link between viroids and viruses is a beneficial pathogen, if you want to call it a pathogen, a beneficial viroid in the oral microbiome and they believe they're replicated inside the bacteria. So yeah, we painted all these viruses as in this Hollywood manner of outbreak and spread fear, and we're running a test and quarantine everybody. But the majority of them are probably mutualistic or providing some type of role we don't fully understand. And I think it does bring us back to this: how do you keep your immune system in equilibrium and homeostasis? Because I think these viruses really only get you when your immune system's out of whack. Low vitamin D in the wintertime, whatever. There's seasonality to them, and otherwise they're probably playing some role we don't appreciate that's beneficial.
Totally. I totally agree. And also this, I mean, kind of to circle back to the idea of like having a narrow band mRNA platform, immediately, like as a molecular biologist myself, I was like, this doesn't make any sense because if we're just targeting specific proteins of this virus, of course that's going to drive viral evolution. Like they're going to create escape variants. Like this seems like common sense. So what
It
is
was
going on
crazy.
here?
On top of that, they threw in mutagens. They threw in
Right.
remdesivir. They threw in
Yes.
molnupiravir, and now there's a whole branch of the Crohn's tree that they've admitted is molnupiravir-induced mutations because they have a certain C to T mutation rate that's different than the virus. And they're like, that's us. We did that.
Is molnupiravir the one that failed the Ames test?
I wouldn't be surprised because I think that's the one that actually affects the mitochondria replication.
Yes.
Yes, yeah.
Yeah, I know there was one that failed the Ames test, and I was like, again, this is just coming back to there's more bacteria than human cells in the body, so what are we doing? Nobody's
Yeah.
thinking.
Yeah, that one I was shocked they got away with, and it didn't look like it had a whole lot of benefit. And then you're going to add a mutagenesis agent in there. You're going to guarantee. I mean, it's a great one-two punch for pharma if they're that smart. That hey, let's convince them of a mutagenic drug on top of a program that's narrowly targeted, and we'll just have a new conveyor belt of new vaccines chasing every single strain out there. And when that doesn't work, we'll throw in Paxlovid. Paxlovid has this problem of rebound.
Mhm.
Anyone who knows what rebound is means the virus is sticking around for more replication cycles, so there's going to get more natural mutation rate going on. One thing after another they used made everything worse, but then more money.
Right, and then there's this talk of, like, wanting to convert the entire schedule over to mRNA, and my head just
Yeah.
explodes.
Yeah. That's when homeschooling gets really serious.
Yeah.
Everyone just get your kids the hell out. I mean, at least the removal of the Department of Education may remove some of the centralization of that type of. There isn't one organization where you could demand vaccine mandates from the head of the government down. But sadly, I don't think removing that's going to change. I think a lot of that zeitgeist is buried in the parents associations and a lot of the schools. That you've got the Karens that are yelling at all the unvaccinated to get their kids vaccinated.
Yep, exactly, and meanwhile, I mean, they're just trusting the science, right? Like, they're not
Yeah.
educated on this, they're just parroting whatever their favorite talking head said, and, yeah, it's just very frustrating, but at the same time, I do feel like there's a lot of shifts happening, conversations being had around these things that used to be kind of taboo to talk about, which is a good thing, and in that way, I'm so thankful for the work that you're doing. I know we've gone quite a long time, so I wanna be mindful of your time, but this was an incredible conversation, I'm so glad that we could do it.
Yeah, indeed, and I'm glad, to put it on a positive note, there are this decentralization technology is here, and that's the message I think we wanna get across is that obviously the media got captured, the journals got captured, everything, they've captured government, right? But people are awake to this now. If there's one thing that happened as a benefit of the pandemic is that a hell of a lot of conspiracists suddenly got, credibility. And people are starting to realize they're being lied to, and decentralization is a term I'm now hearing be thrown around by politicians.
Yep.
So there's an awareness that we need to do this, and it does have to boil down to the money system. The monetary system needs to get decentralized. Once that gets decentralized, it's going to be harder for capture to occur in all these other places. And just by fixing that one thing, it will start to trickle out and infuse, a decentralized mindset. I think in every other industry, it just may take another decade or so. But I've never seen more people aware of this issue since COVID. And like prior to this, you tell people that, oh yeah, there's scientific censorship on cannabis. They'd be like, yeah, go smoke another joint. Whatever. It's all in your head. And then COVID happens, and you can be like, see, I told you, first time.
Yes.
And people are realizing, like, yeah, you're right. Yeah, it is a problem. And this drug war isn't just about drugs that get you high. It's a drug war against anything that's cheap, generic that can leave you free of the medical system.
Yeah, and actually speaking of drugs that get you high, I heard there's some research going on to try to figure out a way to make psychedelics not psychoactive, but still somehow impart their benefits, and I'm just thinking, somehow they're missing the point that there's, like,
Yes,
something
yeah,
going
yeah,
on
yeah.
within the psyche that's changing, that's giving the benefits.
Yeah. There's a good debate on that field, and I don't have a
Yeah.
firm opinion on it. Great, let people explore that. So they're looking for 5-HT, serotonin to a um, agonist that don't trigger the psychedelic effect, and
Yep.
sometimes hit other serotonin receptors. But there may be a therapeutic benefit to hitting those other receptors. I just don't know that
Yeah.
you're going to get this radical. Like the most impressive thing about psilocybin is that you can take treatment resistant depression patients, and 75% of them you can basically eliminate the depression in 2 or 3 therapy sessions, and it's done. Right? It's not lifelong SSRIs, and you might shoot up a school. Right?
Yep.
It is a complete revolution in that field, which is one reason why I don't think it's going to continue to get banned in various places, because the pharma industry is going to find a way to demonize it, like, kids are getting high or whatever. So Florida's talking about banning the spores again, which is like, retrograde again. Um, and so they're going to continue to incite fear from the recreational misuse, if you will, standpoint, so that they can demonize it for getting looked at in where it's going to wipe out their SSRI market.
Yep.
That's how they do. That's how they play this game. And unfortunately, we just don't have anything else like that. And that's a revolutionary compound. I'm of the opinion that you're thinking, that I don't think you get that type of rewiring of the brain without some psychedelic experience.
Yeah.
I think there's something to that, but we'll see where the science falls out on that later in the future. I've not found a control compound that's necessarily. I mean, maybe it's originacin, maybe it's baeocystin, these other tryptamines that are really close to psilocybin, but don't have psychedelic experiences. But I don't know of anyone who's synthesizing those or making those at levels that could where that study could be done. But yes, there are some exciting startups that are in that space. I think Cybin's one of them, and a couple others that are pursuing derivatives of psilocybin that may not have as much of a psychedelic experience that can perhaps hit other depression markets. But I'm skeptical until seeing otherwise.
Yeah, like there could be some neuroplasticity benefits or something like this. The
Absolutely.
other insidious thing about the SSRIs is if you're on them, you can't take psychedelics. They don't work.
Yeah, well, that's another thing about the folks that are tracking methylene blue is keep an eye on that because methylene blue
That's true.
is an MOAI,
Yeah.
and that may make the trip last a hell of a lot longer or push you into sort of a serotonin syndrome
Yep.
situation. So I don't know that it's wise to mix methylene blue with those things, but um, yeah, there's a lot to be learned, I think, on that front, and how to. I know a lot of people who mix them. They do mix harmine with psychedelics. I mean, right? That's how ayahuasca works. Is there's an
Yep.
MOI involved, but it's more of a natural DMT
Right.
that prevents the breakdown of. And I know people mixing niacin and lion's mane, a couple other things to try to change the behavior of psilocybin.
Yeah, I think it's really interesting to think about and a lot of the ancient and like more shamanic cultures will combine different plants together and have this incredible wisdom around the interactions without having to know the exact chemicals involved. It's pretty awesome.
It's amazing. How do they figure that out?
Yeah.
How do you mix two different chemical? I mean, there has to be a lot of experimentation and death. It seems like.
Yeah. Well, actually, it's pretty cool, but I don't remember who I heard say this, but somebody had visited and was speaking to one of the medicine guides down there, and they were saying how, basically there's two plants that you need to combine to make ayahuasca. One has the MAOI and one has the DMT that are in like completely different parts of the rainforest. So like and the way that they described it is that the plants told them where to find them, which is crazy,
Yeah,
right? But like, hey, I'm not going to argue. I mean, it's very possible. There's a lot we
yeah.
don't
I mean,
understand.
yeah, I know. It's like have you listened into those telepathy tapes?
No, but I heard Kai on Joe Rogan's podcast, so I'm, like, tangentially familiar.
Yeah, yeah. I
But
know.
it's amazing.
I'm as new to it as you are, but I hear that, and that's where my head goes. Is okay. That's
Yes.
probably they figured out how to make ayahuasca.
Yes, it's incredible, and this is the beauty of the decentralized approach is, like, we can study these things and we can, like, do it in a way that's not going to, like, rape and pillage the rainforest or, like, actually respecting the culture and just
Right,
making some
right.
understanding that then can be applied to Western culture to make us more, healthy and happy and more functional.
We certainly need to be more open minded about that stuff. It's where has this pillar of science led us where we only trust the white lab coats from Fauci and we shut everything else down, right?
Yeah, it's crazy. It's anti-scientific, really.
It is.
Yeah. Anyway, Kevin, thank you so much for your time. This was extremely fun. Do you want to briefly just tell people where they can find you,
Oh,
your
yeah,
X
sure.
Substack, and also include links to everything?
Okay, so I'm on X as under my name, Kevin McKernan, but most of my writing's done on Substack. So you can find the Substack thing is under nepentolactones. That's the compound that's in catnip, cat person. And sometimes it comes up under anandamide as well, which is we were speaking about that earlier. It's one of the
Endocannabinoid.
endocannabinoids. So you'll find a lot of stuff on there about the cannabis work I do and the vaccine stuff that we stumbled into on Substack. I can only so much of it can go on Twitter because Twitter's it's not perfectly uncensored yet. And then our work at Medicinal Genomics you can find we make a lot of genomic tools for the cannabis space and the philosophy space for helping people make breed better plants and clean plants. And then I think the last one is CanMed. Yeah, we have a conference every year, scientific conference. This year's down in Puerto Rico. It's June, just before the Bitcoin conference in Prague. I'll be at both. So the one in Puerto Rico, I think, just starts on the 16th of June and goes to maybe the 20th, and then I'll be out at the Prague meeting, the Bitcoin Prague meeting on the 21st.
Awesome. Thank you so much. This was so fun. Hopefully we can chat again, offline. I'm going to be up in New England area like sometime during the summer too, so you might be away at your conferences. But anyway, it would be great to connect
But yeah,
sometime in
well, let me know.
person.
We're up in Marblehead, so it's if you make it to the Boston area, we're not too far.
Great. Sounds good. Well, have a great day. Thank you so
All right, you
much.
too. Thank you for the time. Appreciate it. Take care.
Bye.